of action potentials. Now, we know that this is important for all CNS function, including cognition, which is, of course, of relevance for the DRI. We know that myelin is damaged in the process called demyelination, and this is seen in numerous neurological conditions, including multiple sclerosis, Alzheimer's disease, and even in normal aging. And this contributes to nerve dysfunction and has recently also been associated with cognitive impairment. Now, we know that myelin can be regenerated in the process of remyelination, and this involves first the recruitment of oligodendrocyte progenitor cells, or OPCs, that differentiate into new oligodendrocytes that then extend their processes to ensheath the axons. And this remyelination restores axonal health and function, and of relevance to the DRI has also recently been shown to be relevant for cognition. So importantly, we know that this efficiency of remyelination decreases with aging and with disease progression in MS and Alzheimer's disease. Although there are some therapies currently in clinical trials to enhance remyelination, we don't yet know if they work and if they're going to work in everyone. So it's still important to understand the mechanisms that support remyelination in order to identify new regenerative strategies. Now, the first glial-glial interaction I'm going to talk about today relates to microglia, these central nervous system resident macrophages. And work by us and others have shown that these are really critical in instructing remyelination. And our previous work showed that they do this by undergoing a transition in activation and function from pro-inflammatory, which we can identify using a number of markers, including INOS, the transition to a pro-regenerative profile that we can identify using other markers, including arginase 1. And following this transition, these regenerative microglia then support remyelination by enhancing OPC differentiation into mature oligodendrocytes and promoting remyelination. And we identified one of these secreted factors from regenerative microglia that drive this process as being activin A. So we discovered that this transition in microglia activation is one of the rate-limiting steps for remyelination efficiency, because remyelination fails when these pro-inflammatory microglia persist, like we've seen occurs in aging and chronic multiple sclerosis lesions. And we also know that remyelination fails when these pro-regenerative microglia are specifically depleted. So although we know that this transition in microglia activation is key for remyelination, what we didn't understand was how this transition took place. And when we looked in the literature, we realized that there wasn't any direct evidence for a transition in microglia activation in vivo. And so this is a question that was undertook by Amy Lloyd during the course of her PhD with us. And she aimed to really understand how this transition took place in order to identify new strategies for enhancing remyelination. And so what Amy aimed to do was to perform RNA sequencing of these different microglia profiles to better understand the mechanisms controlling their activation. So we use an in vivo model where we induce demyelination in the white matter tract of the adult mouse called the corpus callosum. And we do this by injecting the myelin toxin LPC. So the advantage of this model is that we have a temporal separation between the demyelination and the subsequent remyelination, so that we could really look at regenerative responses in isolation without confounding responses to damage. And in this model, we have a very clear temporal profiling of glial responses over time. So we know that the pro-inflammatory microglia peak at 3 days post-LPC, and the pro-regenerative microglia peak at 10 days post-LPC. So we isolated these microglia at these different time points by FACS. This is just showing how we did this. We isolated the live cells. We then isolated myeloid cells based on expression of CD11b. We excluded Ly6G positive neutrophils and CD3 positive T cells. And then we included microglia based on low expression of CD45 to differentiate them from monocyte-derived macrophages. Now, over the past few years, a microglia gene signature has been established, which includes genes that are either specific or enriched in microglia. And here we're looking at the results of our RNA sequencing with expression of these genes. And you can see that these are expressed by microglia at both of these time points. We also saw that the cells we isolated did not express genes that are associated with other macrophage populations, like the border-associated macrophages or the bone marrow-derived cells, the monocyte-derived macrophages. We did find, however, that our cells express genes that are not expressed by the other macrophage populations, Sigma H and cell 1. So here we're confident that we were sequencing microglia. And then we performed pathway analysis on these RNA sequences. And we found that one of the major molecular and cellular functions that was regulated was cell death and survival. So this was quite surprising to us. So what we did was assess microglia death. First, we looked at microglia numbers. We're looking at IBA1 positive cells. And we found that in between the peak in our pro-inflammatory microglia at 3 days and our peak of pro-regenerative microglia at 10 days, we saw a drop in the microglia numbers. So then we asked whether this drop represented microglia death. We used flow cytometry to analyze all of the microglia within the lesion, and we looked for expression of death markers and XM5 and 7-AAD. And indeed, in between this peak in our pro-inflammatory microglia and our peak in our pro-regenerative microglia, we had an increase in microglia death. So surprisingly, we found that microglia die during efficient remyelination between this transition from pro-inflammatory to pro-regenerative. So we wanted to get a better idea of how this death was taking place. Given that in the brain, the white matter is quite deep, doing live imaging in vivo of the white matter is very challenging. And so we did this in organotypic cerebellar explants that we demyelinate with LPC. And what you're seeing here is a microglia that were imaged based on expression of GFP, as this is driven by the CSF1R promoter. So hopefully what you can see on the left is a video of our control explant microglia that are actively sensing their environment, putting out processes as they should be doing. And on the right, I'll show you a video where we started the imaging about 20 minutes after we added the LPC, and you'll see that the cells are rounding up and disappearing. And hopefully what you can see, if you keep your eyes on the arrows, is that the cells explode, which was quite cool, actually. So we then thought, okay, the cells are exploding. This is not likely to be apoptosis because the cells would implode. And in fact, the cells were negative for apoptosis markers like tunnel and cleaved caspase 3. So we became interested in a type of cell death called necroptosis. So this is a controlled necrosis where this complex called the necroptosome, formed of RIP kinase 1, RIP kinase 3, and an LKL, poke holes in the membrane to cause the cell to explode. And we became interested in necroptosis because it had recently been shown that this controls the change in activation of macrophages in the liver from pro-inflammatory to wound repair following infection. So we wondered whether the same thing could be happening to microglia during remyelination. So Amy stained for necroptosome markers in our in vivo lesions. Here we're looking at RIP kinase 3 expression in CD68 positive microglia and macrophages. And we found that we had a high proportion of necroptosome expressing microglia at 3 days post-LPC prior to when the cells die. So we had engagement of the necroptosome prior to when the cells are dying. Here on the right, I'm showing images of a different necroptosome marker, an LKL, and you can see that these necroptosomes are formed within these IBA1 positive microglia. And so we're seeing that microglia are dying by necroptosis or controlled necrosis during remyelination. So then the obvious question is, why are the microglia dying by necroptosis? What's the influence on microglia and on remyelination? So we blocked this process using a small molecule called necrostatin 1, and this prevents the formation of the necroptosome. We wanted to deliver the necrostatin 1 to microglia, and so we teamed up with Anne Davier, based in Belgium, who created these lipidic nanocapsules, or LNCs, that are essentially these lipid-rich balls that are optimized to deliver drugs. And we hypothesized that because microglia love phagocytosing lipid-rich things, that they would preferentially take up these lipidic nanocapsules. So Anne encapsulated the necrostatin 1 within these LNCs for a vehicle control, and we injected these within the brain at the same time as lesioning to prevent the necroptosis signaling that starts 3 days later. And so we wanted to track where these LNCs were going, so we encapsulated the fluorescent dye, dye-D. Here where you're seeing these white clusters is where the LNCs have been taken up by cells. And so here you can see that these dye-D clusters were associated with IBA1 positive microglia. And here we did a 3D rendering where you can see that these dye-D clusters are inside the IBA1 positive cells. Now, we then assessed the percentage of the dye-D clusters that were co-localized with cell types that are known to phagocytose. So although we know that microglia and macrophages have the highest phagocytic potential within lesions, we also know that astrocytes and oligodendrocytes can also phagocytose. So we looked at the percentage of dye-D clusters that were co-localized with these different cell types, and we found that over 90% of the clusters were associated with IBA1 positive microglia, and only a very small percentage with GFAP positive astrocytes or CC1 positive oligodendrocytes. So having delivered the necrostatin to the microglia, we then looked to see whether indeed it inhibited necroptosis. Here we're looking at the necroptosis marker NLKL in red in the IBA1 positive microglia, and you can see these necroptosomes that are localized within the cells in our vehicle LNC control. And when we delivered the necrostatin 1, we had a reduction in these NLKL positive microglia. So then we looked at the microglial responses. This is normalized over vehicle LNC control. And first we looked at total numbers of microglia using TNEM119 as our marker, which will mark all microglia regardless of their activation. And we found that we had a twofold increase in the number of microglia. So here we really prevented the death of the microglia, and we confirmed that this did not represent proliferation of the cells. Then we looked at the activation of the microglia, and we found that there was a higher proportion of pro-inflammatory microglia that we could detect with our marker INOS, and we had a reduction in the pro-regenerative microglia that we detect with our marker arginase 1. So this really showed us that blocking microglia necroptosis causes persistence of these pro-inflammatory microglia and prevents the emergence of these pro-regenerative microglia. So the necroptosis is really required for this transition inactivation to take place. We then looked at the consequence for remyelination. Here we're looking in the corpus callosum, outlined in white, and we're looking at the early remyelination marker MAG, this myelin protein. And you can see a very robust remyelination in our vehicle LNCs, but in our necrostatin 1 delivery, you can see that this is impaired. So we measured the MAG mean pixel intensity, and compared to our vehicle control, we significantly impaired remyelination when we inhibited necroptosis in the microglia. So blocking microglia necroptosis inhibits remyelination, and so microglia necroptosis is required for remyelination. We then looked at the translational relevance of our findings. We looked in multiple sclerosis tissue. Here you can see a tissue block where the myelin has been stained for voxel-fast blue here. And the lesions are outlined in red where we have less blue because there has been demyelination. We looked in different lesion types that have different remyelination potential. Active lesions have high remyelination, and chronic inactive lesions, which have been around for a long time, have poor remyelination. We looked at the necroptosis microglia, looking at CD68 MLKL double positive cells. These were rarely detected in healthy controls, but they were increased in our active lesions that have high remyelination, and less so in chronic inactive lesions that have poor remyelination, or lesions that had already fully remyelinated. And so here are some images on the right where we're seeing RIP kinase 3 positive CD68 double positive cells that are present in these active lesions, but not in our control. So even in human tissue, we were able to associate microglia necroptosis with high remyelination potential. So I've shown you that during remyelination, we have death, these pro-inflammatory microglia, but we know that they come back. They repopulate to this regenerative profile. So we wanted to assess this, and work in recent years has identified that following experimental depletion of microglia, when they repopulate, they express the marker nestin. And so we looked at nestin expression first in our mouse model, and at 7 days post-LPC, at the same time when the microglia are dying, we're seeing IBA1 expression in the nestin expression in the IBA1 positive cells, and this is the merge group. We also looked in our multiple sclerosis lesions, where we looked at nestin expression in our P1 positive microglia, and we found that these were not expressed or not detected in control tissue, but these were increased in active lesions that have high remyelination and less so in chronic inactive or fully remyelinated lesions. So we're seeing this microglia repopulation marker occurring in association with effective remyelination. We became interested in the molecular mechanisms that could control this. So we went back to our RNA sequencing and the pathway analysis, and we found that interferon signaling came up quite a bit. And so more specifically, this was type 1 interferon signaling, which involves interferon receptor 1 and interferon receptor 2 that form this complex. And downstream, you have activation of STAT1, which is phosphorylated. And so the first thing we wanted to do was check whether our microglia were expressing the interferon receptor during remyelination. So we looked at 3, 7, and 10 days at our CD68 positive cells, and indeed we found that they expressed interferon receptor 2. We then looked at activity of this signaling by looking at phosphorylation of STAT1, which is normally found within the nucleus. So here we looked at microglia nuclei using PU1, shown here in magenta. And what you can see is that we only had phospho-STAT1 in microglia nuclei at 7 days post-LPC. And if you zoom out of this image, what we found is that it was really only a subset of the microglia that had activation of interferon signaling. We had phospho-STAT1 positive, PU1 positive nuclei in the center of the lesion, and then we had some PU1 positive nuclei that were negative on the outside. So this was really interesting to us because it was occurring at 7 days post-LPC, when we know that we have both microglia death and microglia repopulation. So we wondered whether the interferon signaling was regulating one of those processes. Is it expressed in the dying microglia, or is it regulating the repopulation? And so this was a little bit challenging to assess in vivo because everything's happening at the same time. And so we went back to our explant cultures that I introduced earlier, because we had seen that in these explants, we have a better temporal separation between the microglia death and the repopulation. So I'll just summarize this here. Here we're looking at the number of microglia over time post-LPC. And in black, we're seeing the total number of microglia expressing CD68 that really peak at half a day after LPC. And the majority of these cells are pro-inflammatory at this time, expressing INOS. Then we see that the microglia die by 1 day, they disappear. And then they repopulate by 7 days, and all of the cells are expressing a regenerative marker, arginase 1. And so we're seeing this really nice separation between the peak of death and the peak of repopulation. So what we did was block the interferon signaling within the slice cultures and looked at death and repopulation of the microglia. We did this by using a neutralizing antibody against interferon receptor 2 that blocks the downstream signaling. And we compared this to an isotype control that does not bind interferon receptor 2. So we looked at microglia nuclei, and at 1 day at the peak of microglia death, we didn't see any difference between the conditions. But we did see that there was an increase in microglia numbers over time in our control, which indicates repopulation. And we saw that this was aggregated when we blocked the interferon signaling. So we basically prevented the microglia repopulation by blocking interferon signaling. These are the images on the right with the microglia nuclei indicated by P1 in magenta. In our isotype control, this is the repopulation, and you can see that we had much less repopulation with the interferon receptor blockade. And so this is showing us that blocking interferon signaling impairs microglia repopulation, and so interferon signaling is needed for microglia repopulation. So then we asked, what's the impact on remyelination? And so we let these explants go for a little bit longer. We used our neutralizing antibody technique, And this is our isotype control, where we have nice remyelination that started. This is the myelin protein MVP, and you can see that this has nice colocalization with our axonal marker neurofilament. But when we blocked interferon signaling, we had an impairment in remyelination, even though the axons are still there. And so we've measured the remyelination, and you can see that comparing the isotype control to our interferon blockade, that we've impaired remyelination. So the interferon signaling is needed for remyelination to take place. So as a summary of this part of the talk, what we've shown is that this transition in microglia activation from pro-inflammatory to pro-regenerative that is needed for remyelination to take place occurs by death from necroptosis and then repopulation via interferon signaling. And so this raises three important points. The first is that microglia death and repopulation regulate remyelination. We've also seen, or this would suggest, that remyelination failure may reflect disrupted microglia turnover. And it raises the provocative question, could we enhance the death of pro-inflammatory microglia as a therapeutic strategy to promote central nervous system remyelination? So this is what's being addressed by our PhD student, Aisha Mahmood, who has a poster at this meeting. And she's developed a really beautiful in vitro pipeline for drug screening to assess the impact on microglia death, comparing control microglia to those we've activated to a pro-inflammatory profile. She's optimized automated counting, and here's just an example of some preliminary findings where she's normalized cell count using Z-score. And here we're basically looking at the number of microglia in our different conditions. And the lower the Z-score, the less microglia we have. We have our positive controls for killing cells here in orange, and then she's already identified some hit compounds in red that kill microglia in our pro-inflammatory conditions. And so I'd like to now move on to the second part of the talk, where we're looking at another glial cell type, the astrocyte. And this project is being led by Irene Molina-Gonzalez. She started this during the course of her PhD and has continued on as a postdoc, and you'll hear from her later today during the panel discussion, because she's developed some really cool in vitro systems to investigate astrocyte-oligodendrocyte interaction. So when Irene started in the lab, she was really curious about how astrocytes regulate remyelination. And the role of astrocytes in remyelination is really unclear and controversial. They've been shown to both promote and inhibit OPC proliferation, promote or inhibit oligodendrocyte differentiation, and promote or inhibit remyelination. And so although astrocytes are the most abundant cell type in demyelinated lesions, we still don't know how to target them to enhance remyelination because we don't understand what they're doing and what regulates their function. And so what Irene did first was characterize the astrocyte responses during remyelination, here again using the LPC model. And she looked at different key time points during remyelination, at 3 days post-injection when we have our OPC responses, at 7 days post-injection when we have these new oligodendrocytes being generated that then need to survive, to then produce the new myelin that occurs between 10 and 21 days post-injection. And so here, Irene looked at the reactivity marker, GFAP, in the corpus callosum outlined in white, and you can see that over the course of remyelination that we have an increase in astrocyte reactivity. And this is really peaking at the time when we know we have new oligodendrocytes being formed and remyelination is starting. So that suggests that they have a role in remyelination. And Irene wanted to better understand what that role could be. And so what she did was sequence the translatomes of the astrocytes. So what this means is really isolating and sequencing mRNAs that are being actively translated in astrocytes. It's kind of a step up from RNA sequencing in that you're sequencing mRNAs that will go on to make protein. So she did this specifically in astrocytes by trap sequencing using transgenic mice in which ribosomes in astrocytes have a GFP tag. And this is because the ribosomal subunit is, the GFP tag to the ribosomal subunit is driven by the pan-astrocytic promoter ALDH101. And so Irene could then isolate the astrocyte ribosomes based on GFP expression and then sequence the mRNAs that are associated with these ribosomes. On the right is a proof of concept of the trap sequencing. where compared to the non-isolated RNA, we've been able to see an enrichment of the trap sequences with astrocyte-associated genes, shown here in pink, and we don't see enrichment of genes associated with neurons, oligodendrocytes, microglia, or endothelial cells. And so Irene then looked at these translotomes at these different time points, and here we're looking at plots looking at the log 2-fold change compared to p-value, where the upregulated genes are shown in red and the downregulated genes are shown in blue. And you can see that we have changes in these genes at all 3 time points, but interestingly, when looking at the top genes that are regulated, they were different at every time point. And so this suggested that there were changes in astrocyte function at different time points. And so Irene then carried out pathway analysis in our astrocyte translotome sequences, and she started at 3 days post-injection at this early time point. And we found that the astrocytes had an NRF2 profile. So NRF2 is an antioxidant pathway, and NRF2 itself regulates the expression of target genes that are involved normally in protective responses. And so here we're looking at a heat map of NRF2 target genes, and these were upregulated at 3 days post-injection compared to our control. And in fact, we found that these went down over time by 7 days. Here we're looking at 7 days versus 3 days. You can see they're then downregulated. So we have a transient activation of the NRF2 pathway in the astrocytes. And Irene then validated this. Here we're looking at the target protein HMOX1 in magenta in the astrocytes, expressing GFAP. We looked at no lesion and sham injection controls, and you can see that at 3 days post-injection that we have an upregulation of HMOX1 expressing GFAP positive astrocytes. This is quantified here, and you can see compared to the no lesion or the sham controls that we have this transient increase in NRF2 activated astrocytes. Irene then went on to look at the later time point of 7 days when these new oligodendrocytes are being formed, and she found that the cholesterol biosynthesis pathway was engaged. And so here we're looking at cholesterol biosynthesis pathway genes in this heat map compared to the no lesion control, and these are upregulated at 7 days post-injection. Previous work has shown that astrocyte translotomes in the poorly remyelinating model EAE have a decrease in the cholesterol pathway, and here we've included this side by side so that you can see that this is down when we have poor remyelination, but it's up when we have efficient remyelination. And indeed, we found that this was up compared to 3 days. So at 7 days, we had an increase in these cholesterol pathway genes. This was validated here. We're looking at the cholesterol biosynthesis pathway enzyme, HMGCS1. Compared to no lesion and sham controls, at 7 days, we have this increase in HMGCS1 positive, GFAP positive astrocytes. And this is quantified here compared to the no lesion and sham controls. You can see this increase in the cholesterol pathway activated astrocytes at 7 days. So what I've shown you so far is that during remyelination, astrocytes have this reciprocal activation of the NRF2 and cholesterol pathways, where NRF2 comes up first, then it goes down, and then cholesterol comes up. And Irene found something interesting in the literature where in the liver, NRF2 downregulation leads to cholesterol upregulation. So we wondered if this downregulation of the NRF2 was needed in order for the cholesterol to come up. And we thought, why don't we stop the NRF2 from being downregulated and see what happens? And so we did this by using a transgenic mouse in which the GFAP promoter induces expression of NRF2, and this overactivates the NRF2 pathway. And so the astrocytes from this mouse were sequenced, and you can see here the NRF2 gene is up, as are NRF2 target genes compared to wild-type astrocytes. So then we asked, could the sustained activation of the NRF2 pathway block the cholesterol biosynthesis pathway and have an impact on remyelination? So using these GFAP NRF2 mice, the first thing that Irene saw was that indeed there was a reduction in cholesterol pathway activation. Looking at the top panel, we have the GFAP positive astrocytes that are expressing the cholesterol biosynthesis enzyme HMGCS1, but in the GFAP NRF2 mice, you can see that this is decreased. We then found that this was linked with poor remyelination. So first we confirmed that in our GFAP and our 2 mice, we had normal myelination to begin with without lesioning. And here we're looking at the myelin protein MVP in the corpus callosum, and you can see that the staining is similar between the wild type and the GFAP and our 2 mice. But once we've demyelinated, we have robust remyelination in our wild type mice, but this is impaired in the GFAP and our 2 mice. And this is quantified here by looking at the percentage MVP area. No difference when we have no lesion, but when we've lesioned, you can see that the wild type has higher remyelination compared to our GFAP and our 2 mice. So this sustained NR2 activation in astrocytes led to impaired remyelination. So Irene then went to understand why we saw poor remyelination in these mice. She looked at mature oligodendrocytes that express CC1, and we used olive2 as our pan-oligodendrocyte marker. So there was no difference in our no lesion controls, but following demyelination, we had a reduction in mature oligodendrocytes in these GFAP and NR2 mice. So then Irene found that this was because of apoptosis of oligodendrocytes. She looked at cleaved caspase-3 expression in the oligodendrocyte lineage, and she found that at 7 days post-injection, before we lose the cells, that we had an increase in apoptotic oligodendrocytes in these GFAP and NR2 mice. And so we wanted to better understand how this sustained NR2 activation in astrocytes and decreased cholesterol production could lead to poor survival of oligodendrocytes. So we went back to our old friend Amy Lloyd, who's now a postdoc in Dundee, and who specializes in proteomics. So we thought, okay, let's send her lesions and do proteomics on, and to see if we could identify what, in relation to the cholesterol pathway, could be related to what we're seeing in the mice. And so doing Bayesian proteomics, we see that we have upregulation of cholesterol biosynthesis pathway genes at 7 and 10 days post-injection. And the most highly upregulated of these proteins is ABCA1. So this was really of interest because ABCA1 is a cholesterol exporter. So it regulates cholesterol efflux. And what's interesting is that it's known that ABCA1 is most highly expressed by astrocytes. And so we looked at ABCA1 expression in astrocytes during remyelination, and we can see that compared to the no lesion control, we have an increase in ABCA1 expressing astrocytes, particularly at the time when we have these new oligodendrocytes being generated that are vulnerable to cell death. And we looked at the proportion of the ABCA1 positive cells that are astrocytes, and we found that this was the majority of the cells. So we thought, okay, ABCA1 is useful in, or used for cholesterol export. Could we target ABCA1 to restore function in our GFAP and our 2 mice? And so we did this by using CS6253, which is an ABCA1 agonist. And interestingly, it not only promotes cholesterol export, but also expression of cholesterol biosynthesis pathway genes. And this has been used previously in EAE to restore the cholesterol pathway in astrocytes. And so Irene applied the CS6253 to our GFAP and our 2 mice between 7 and 14 days post-injection, when we know in the wild type that the cholesterol biosynthesis pathway should be activated, but when we know that it's not being activated in these mice. And so indeed she did activate the cholesterol pathway in these cells. We're looking at HMGCS1 expression in astrocytes, and you can see compared to our PBS control that we've increased the proportion of astrocytes that have activation of this pathway. And what we found was that the CS6253 application decreased the apoptotic mature oligodendrocytes, CC1 positive cells. Compared to our PBS control, this was decreased. And this resulted in restored remyelination. So we're looking at the myelin protein MPP in our GFAP and our 2 mice. This is poor in our PBS controls and restored in our CS6253 treatment, quantified here, percent MVP area, comparing the PBS to the CS6253. So here we've been showing that restoring cholesterol biosynthesis pathway activation when the NRF2 activation is sustained in astrocyte rescues remyelination. So then Irene asked whether astrocytes directly transfer cholesterol to oligodendrocytes to regulate their survival. Although this is something we had inferred in vivo, it never actually had been directly shown. And so Irene came up with a clever way of doing this by applying a fluorescently labeled cholesterol, that's BODIPI labeled, loading that in the astrocytes, and then allowing astrocytes to efflux this into the astrocyte conditioned media. Then we applied this astrocyte conditioned media to mature oligodendrocytes. And what Irene asked was, if she manipulated the NR2 pathway, could she influence this transfer of cholesterol to mature oligodendrocytes? So she activated the NR2 pathway in the astrocytes using a potent activator called CDDO. She then used as a control an NR2 inhibitor, luteolin, that would then inhibit the NR2 activation to restore it back to normal. And she also used these CDDO activated astrocytes to then correct the cholesterol pathway using the CS6253 that I just presented to you. And so what we found was that the oligodendrocytes did take up the labeled cholesterol from the astrocyte conditioned media. This is our astrocyte media control without any conditioned media. And these are our mature oligodendrocytes here expressing TPPP, P25, and SOX10. And you can see these BODIPI positive oligodendrocytes when we use the astrocyte conditioned media. And this was actually increased when we had the CDDO luteolin conditions or the CDDO C66253 condition, showing that if we enhance if we inhibit NR2 or if we enhance the cholesterol pathway, that we can increase cholesterol transfer to oligodendrocytes. Irene then linked this to oligodendrocyte survival. She looked at apoptotic oligodendrocytes by looking at tunnel-positive TPPP, P25 positive cells. And here we're looking at the data normalized to our oligodendrocyte media controls. And when we added the CDDO astrocyte conditioned media, the death was increased, but this was rescued when we had either blocked NR2 using luteolin or stimulated cholesterol using CS6253. So here Irene's been able to show that astrocytes directly transfer cholesterol to oligodendrocytes to regulate their survival. NRF2 negatively regulates this, and you can restore this by either blocking NRF2 or stimulating the cholesterol pathway. Then Irene did an experiment where she wanted to understand whether the cholesterol efflux from astrocytes was required for remyelination. She used an ABCA1 inhibitor called PSC833, and this inhibits the export of cholesterol from astrocytes. And so we did this here in remyelinating brain explants, and so we applied the LPC at 2 weeks in culture, and then we waited for the demyelination to be complete, and then we applied the PSC833. And what you can see quite clearly is in our vehicle control, we have robust remyelination shown by MVP expression along the axon shown by neurofilament, and this was inhibited when we blocked ABCA1. And so inhibiting cholesterol export inhibits remyelination. Okay, so then we wanted to look at the translational relevance of our findings. Irene had shown that we have this reciprocal activation of the NR2 and cholesterol pathways in our GFAP NR2 mice. So we took advantage of an existing single nuclei sequencing database of distinct MS lesions where they looked at active and inactive lesions. And we reclustered the astrocytes. And here we're looking at a t-SNE plot where every dot represents a different astrocyte, and they've been clustered together based on similar gene expression shown by distinct colors. And we discovered that there are 8 different clusters of astrocytes, and these were different when comparing control to the MS lesions, and they're also different when comparing active to inactive lesions. And interestingly, we found that in these inactive lesions that have poor remyelination potential, that there was this cluster 1, which was primarily detected in these inactive lesions shown here in pink, more so than in active lesions in blue or control in green. And so analyzing this cluster 1, we found that they had upregulation of NRF2 genes compared to the other clusters, and these are classical NR2 target genes. And we found that they had downregulation of cholesterol genes, including ABCA1, which I previously mentioned. And so what we've seen here is this reciprocal regulation of NRF2 and cholesterol in MS astrocytes, and interestingly, in poorly remyelinating lesions, we have NRF2 that goes up and cholesterol that goes down, which is exactly what we've seen in our GFAP and our NRF2 mice. And Irene is able to validate this at the protein level. Then we looked at whether this was associated with oligodendrocyte death in the MS lesion. So again, we looked at these different lesion types. We looked at mature oligodendrocytes, DPPP, P25 positive, oligodendrocyte lineage cells, OLIG2 positive, that were cleaved caspase 3 positive. And we found that these were highest in these chronic and active lesions that have the high NRF2, low cholesterol, compared to either active lesions or fully remyelinated lesions or control. So we've been able to associate the dysregulation of the NRF2 and cholesterol pathways in MS with oligodendrocyte death. And so lastly, Irene wanted to see if we could identify a drug that could restore remyelination when we have this dysregulation of NRF2. And so we went back to luteolin, which I introduced in the in vitro studies, which has been used to correct NRF2 when it's dysregulated, when it's hyperactivated. And this was applied between 4 and 7 days post-injection in vivo, during the time when NRF2 is normally downregulated, but when it's not downregulated in our GFAP and NRF2 mice. And so Irene was able to confirm that the luteolin reduced NRF2 activation in astrocytes, and she also saw that it enhanced the cholesterol pathway activation in these cells. And really excitingly, what she found was that this reduced oligodendrocyte death. So these are the OLIG2 positive, cleaved caspase 3 positive cells. Compared to our vehicle control with the luteolin, we've decreased oligodendrocyte death. And this led to improved remyelination. We're looking at the myelin protein NPP in purple. You can see that this is poor in the PBS control, but rescued with the luteolin. And this is the quantification of the percentage MVP area, where with luteolin, we've restored remyelination. So this is really exciting that we've been able to rescue remyelination in the GFP and NRF2 mice using an existing drug. And so as a summary of this part of the talk, what Irene discovered was that during remyelination, astrocytes undergo a switch from NRF2 activated to cholesterol activated, and this drives the survival of the oligodendrocytes. And which has also been able to show is that this downregulation of the NRF2 allows cholesterol biosynthesis and efflux to support the survival. And importantly, this highlights astrocytes and mature oligodendrocytes as key therapeutic targets for remyelination, beyond just driving OPC differentiation, which is the current favored strategy that's being tested in clinical trial. And so to put these two parts of my talk together, what we've seen is that during remyelination, that we have a transition in microreactivation from pro-inflammatory to pro-regenerative that drives the differentiation of the oligodendrocytes, and this occurs via cell death. And then the astrocytes switch from NRF2 activated to cholesterol pathway activated that then allows the survival of these newly generated oligodendrocytes. And so these glial-glial interactions are required to regulate central nervous system remyelination. And so I'd just like to thank my wonderful lab who did this work. So the first part of the study was done by Amy Lloyd, who's now a postdoc in Dundee. She was supported by Claire Davies, who was a postdoc in the lab who did the RNA sequencing, and was also supported by our research assistant, Rebecca Holloway. The astrocyte story was carried out by Irene Molina Gonzalez, an absolute superstar who you'll hear about later, and she was also supported by Rebecca and Aisha and Lindsay in her study. I'd also like to point out that we have another poster at the meeting by Neat McNamara, who's looking at how microglia regulate myelin integrity. And so I'd like to thank our collaborators both in Edinburgh and beyond, and our funders. And I'd like to thank you for your attention, and I'm happy to take your questions.
That was really good, Veronica. Just a few bits of data. So does anyone have any questions? Go on, Quiva.
Do I just speak it?
Can you hear that, Veronica? Can you hear Quiva?
Yeah, I think she'll have to speak a little bit loudly, but I can hear her.
Okay, I can try and speak loudly. Sorry, that was really interesting. I suppose I'm just interested in the NRF2 stuff in like in the context of stroke, NRF2 is thought to be neuroprotective, and things like certain drugs that are activating NRF2 are thought to be beneficial. So just curious what your thoughts are in terms of MS. Do you think that would make everything worse in the context of MS, or do you think if you activate NRF2 as long as you get that cholesterol biosynthesis up, that it could be beneficial overall, or that you really need to inhibit it in context of myelination?
Yeah, that's a great question. So we know that, and there's so much evidence out there, that NRF2 is protective against demyelination. It's been shown in numerous different types of animal models, including in EAE and in stroke, as we mentioned. And so we weren't able to say a protective effect of NRF2 against demyelination because we think it was a response to the demyelination. But interestingly, it's never been specifically shown with these NRF2 activating drugs. So, for example, Tecfidera, which is being given to people with MS, it's never been shown the direct impact on remyelination. Even in studies where they say they're talking about remyelination, they're using it in a model where you have concurrent demyelination. And so it's probably mostly been a protection against demyelination. And so we think that the downregulation of the NRF2 will be really important in we really need to look at this in the context of progressive disease. And so stimulating the cholesterol pathway, like you said, could be a strategy to try to overcome this sustained NRF2 activation or dysregulated NRF2 activation. And so it's a great question. It's really quite unclear about these drugs, exactly what they're doing in humans, and exactly what they do to remyelination. So we do need to, I think, investigate what they do for remyelination. They're not entirely specific for NRF2 activation. They activate other pathways as well. And so I think it's a great question and something that we need to address.
Thank you.
I had a question, Veronica, regarding the kind of astrocyte transfer of cholesterol to oligodendrocytes and the kind of promotion of survival. Do you know like the exact mechanisms? Have you ever thought of kind of doing metabolomics or lipidomics, like what the kind of transfer of cholesterol, why the astrocytes surviving?
Yeah, so we were interested in doing lipidomics in vivo, but we basically, based on how small the lesions are, it wouldn't be feasible for us to isolate the astrocytes and to be able to really do lipidomics there. It could be something that's possible in vitro, but we haven't explored that yet. We have struggled with measuring cholesterol directly in the conditioned media because there's high serum. We've got the cells in serum, and it basically masks it. So this labeling technique that Irene used was quite a clever way to see that cholesterol itself is effluxed. In terms of metabolomics, that's interesting. Irene has seen some metabolic pathways that seem to be regulated later on in the astrocytes and remyelination at 10 days, but it's not something that we've investigated yet. So far all we know is that we do have efflux via ABCA1. This is taken up by the oligodendrocytes, but exactly what's going on the oligodendrocyte side, we don't know that yet.
Cool. Any other questions?
We've got one from the chat.
There's one on the chat.
Yeah. Jamie's asked, with regard, sorry, can you hear me?
Yes.
Oh, great. So with regards to microglial necrosis, is interferon production increased by LPC? And does interferon signaling promote cell death or repopulation?
Sorry, so is the question whether when interferon itself is upregulated?
So he asks, basically, does interferon production increase LPC? And then he also goes on to ask, does the interferon signaling promote cell death or repopulation?
Right, so that's a really interesting question. We don't yet know where the interferon is coming from. One hypothesis could be the astrocytes, because we have seen an interferon signature coming up later in remyelination, but we don't yet know where and exactly when. In terms of interferon itself, we supplemented with interferon alpha or beta to see whether or not we could kind of drive the repopulation. That's an interesting idea. We haven't done that yet. It's interesting because there is a drug approved for MS that is interferon beta called betaferon, and I'm curious to know whether or not that could influence the repopulation. However, beta interferon does not cross the blood-brain barrier, so we'd have to deliver it, via another mechanism like nanoparticles, for example.
Yep, at the back.
If you can hear me, does ABCA7 transport any other lipids than cholesterol? I know ABCA7 does, but I'm not so sure about ABCA1.
Did you hear that, Veronica?
No, can you repeat that for me, please?
So,
Or maybe Stefan, you can say it, because I think
yeah,
it's
so
hard
what
to
was,
hear.
can you repeat the question, sorry?
It was about whether ABCA7, ABC, A1, does it transport any other lipids than cholesterol? Could there be signaling molecules? ABCA1.
ABCA1, does it transport any other lipids? Is that right?
Yeah.
Yeah, good.
Cholesterol.
I had one job.
Yeah, that's a good question. We don't know. We haven't asked about cholesterol pathway intermediates. It has been shown that microglia release cholesterol pathway intermediates, and that regulates OPC maturation during remyelination. That's not something that we've looked at yet, but we do know that cholesterol itself is transferred based on the OTP experiments. But it's a great question. There could be other intermediates that are important that we haven't looked at yet.
Oh, yep, at the back.
I'll have to shout as well, I'm at the back.
Okay.
Is there any evidence that microglia regulate oligodendrocyte survival or death?
So, is there any evidence to suggest that microglia regulate oligodendrocyte survival or death?
I haven't seen that. We're kind of thinking now that there's a little bit of a tag team going on between the microglia and the astrocytes, so the microglia are more important earlier on to induce OPC differentiation, and then the astrocytes kick in to make sure that those oligodendrocytes survive. Basically, we would need to do more specific microglia depletion experiments to look at the impact on oligodendrocyte survival. Up until now, that's been a little bit challenging because all the tools, including the ones we've used, would have also targeted border-associated macrophages like the perivascular macrophages, which are present within the white matter. We do have this new tool with this new transgenic model where microglia are specifically lacking in the central nervous system that, if you're interested, you can check out the Miron's poster. And we had some very preliminary studies where we looked at remyelination there, and it would be interesting to look at oligodendrocyte survival there. But so that's a long answer to say we don't yet know, but my inclination based on the literature is probably they're more important earlier on.
Yep, Bhuvnesh.
Veronique, good talk around the oligodendrocyte and demyelination. Could I ask you the first bit? Do you know what signals are required for the death of oligodendrocyte during demyelination?
So is the question what induces microglia necroptosis?
Exactly.
Yeah. We don't know. So we have some hypotheses that it could be via TNF receptor signaling. So it is a fairly newly discovered type of cell death. Maybe about 10, 15 years ago it was discovered. And so we're still figuring out the receptors that are involved in driving necroptosis. The TNF receptor 1 is the one that's been most heavily implicated in driving necroptosis. We know that the pro-inflammatory microglia are making lots of TNF and they express high levels of the receptor. So that's one possible explanation that they're kind of driving their own death. Another possibility could be phagocytosis of myelin debris, as that's really what's happening at the time when the pro-inflammatory microglia are starting to engage the necroptosis pathway. And there has been association of TREM2 with necroptosis, and TREM2 is important in myelin debris phagocytosis as well. I know it does a lot of other interesting things, but these are some ideas that we're thinking about that need to be tested.
No questions?
Another one from the chat.
Cool.
Colin asks, astrocytes contain myelin debris in demyelinated lesions. Do you think there is any sort of recycling of these lipids to provide cholesterol to remyelinating oligos?
Yeah, that's a good question. So there was a recent paper in Nature Neuroscience showing that microglia do this. That's something that we've not looked into. We do know that they have activation of the pathway itself. So it wouldn't just be taking up cholesterol and then recycling it back out. So it could be a combination of both, but we haven't investigated that that option of recycling. We do know that for the most part, most of the myelin debris phagocytosis is being carried out by microglia and macrophages. Now, and although astrocytes can do it as well, they're not the primary cells that are doing it. So I would think that it wouldn't be as big of a contributor as making the cholesterol in the astrocytes.
Can I ask another question?
Go for it.
Is that allowed? Sorry, just in terms of the methods, just really quickly, why, just curious to know why you use the cerebellar explants instead of other parts of the brain, and like, would you be ever worried slightly that what you're seeing in the cerebellum might be different? You know, we know the microglia there might be more active, more metabolic, whether it's always going to correlate.
Yeah, to be completely honest, the cerebellar explants are the most established brain explant model in terms of demyelination and remyelination, and so to avoid establishing a whole new model, that's the one that we use. Mm-hmm. So we did see the same process of, necroptosis, death and repopulation as we see in the corpus callosum. Now the time course was different, as you noted, and so we don't know if that time course is because the microglia are activated a bit from the culture anyway. So we do leave them for a couple weeks before we demyelinate them to let them calm down a bit, but they still are activated because they phagocytose quite a lot from the next section. So that could be a reason why we kind of sped up the whole process, or it could be region dependent. So it would be interesting to look at whether there's a difference in the turnover of microglia during remyelination in different brain regions. It is thought that there is a slower remyelination time course in the cortex compared to the corpus callosum, but comparing different white matter tracts, that hasn't really been looked at, especially not in terms of microglia turnover. So it's a good question. Regional differences in microglia responses and remyelination rate are of interest. We haven't looked into that very carefully. We do have a forebrain brain explant model set up now with corpus callosum that we used for perinatal brain injury experiments, but we haven't yet use those for DN remyelination. So that could be one way to address those issues.
Cool, thank you.
Cool. Right, well, in best interest of time, we'll wrap up there. So thank you again, Veronica. That was a really good talk. Cheers. Okay. So I think now we're going on to ECR talks, which Tom is chairing. So.
So much better to have all the tools up and then not have to yeah, switching phones. Yeah, it'd be a nightmare. Yeah, I'm okay. It's pretty fun. Yeah.
You know, you think about really good questions.
Yeah.
Okay, everyone. So we're going to do our first one of ECR talks now. So these are just quick 15-minute talks. We've got a couple minutes of questions afterwards. So starting off, we have Emily from Cardiff.
Cool. So hello, Emily. And so I work in Julie Williams' lab at Cardiff, but before this I worked in Phil Taylor's group in the DRI at Cardiff, and that's when I did this work. So my project aimed to find out how the R522 variant in the enzyme PLC gamma 2 reduces an individual's risk of developing Alzheimer's disease. So what is PLC gamma 2 and how could it be involved in Alzheimer's disease? So my project was started when this paper here was published, and this is a GWAS study where they found that the R522 mutation in PLC gamma 2 is protective against Alzheimer's disease. So in this Manhattan plot here, the SNP is above the significance threshold. So this diagram just shows how PLC gamma 2 works in cells, basically. And so, as I said, it's an enzyme, and it breaks down at this substrate here, which is a phospholipid called PIP2, and this produces IP3 and DAG. So IP3 goes to the IP3 receptor on the ER, and this causes a large increase in cytosolic calcium. And this cytosolic calcium increase triggers a lot of events within the cell, including nitric oxide, cytokine and chemokine release, phagocytosis, and also receptor signaling events. So DAG goes to the nucleus and activates MAP kinase and NF-kappa B signaling pathways. So as an enzyme, PLC-gamma2 represents the first classically druggable target to emerge from late-onset Alzheimer's disease studies. It's primarily expressed in the brain within microglia and macrophages, which is, what we're interested in for the most part. So it lies downstream of TREM2, as you can see here, and we all know everybody loves TREM2. And also in this study here, it was found that the R522 variant has increased enzymatic activity in COS7 cells. So that was quite interesting. Okay, so the first thing that my lab did in order to study this was to use CRISPR to generate iPSCs, or induced pluripotent stem cells, containing the R522 variant and also PLC-gamma2 knockouts as a control, although the data on this slide is just the R522. So the first thing that we did, we generated the R522 and then confirmed that the mutation had been introduced via sequencing, as you can see here. And then I differentiated these cells into microglia, so using a protocol described in Hainzeller et al., whereby basically you just add growth factors to the stem cells, and this causes them to go towards mesodermal lineage and form embroid bodies, and then you add more growth factors to the embroid bodies, and this makes them start to produce these myeloid precursor cells. So these cells, we showed that they have equal expression of PLC-gamma2, and also that they express characteristic myeloid markers, so CD11b, CD14 and CD45, but not CD34 because that's a hematopoietic stem cell marker. And then, so after that, I plate the cells down in astrocyte condition media, which contains what the cells need to complete their differentiation into microglia. And here you can see that they express IBA1, TMEM119, and GLUT5, which is good. And so, broadly speaking, the R522, the control, and the knockout all kind of turn into microglia in a very similar way, which acts as a good kind of baseline for investigating the function of this R522 variant. So, along with my models, so I work with Tom, who's there, and and he made, he didn't make them, the Jackson laboratory made some PLC-gamma2 R522 knock-in mice, and then he also had some APP knock-in mice, which is kind of representative of familial Alzheimer's disease, and then he crossed the R522 with the APP knock-in. And from these models, he generated microglial and macrophage, so MOP lines, which we could use in cell experiments. So the first thing that I looked at was I wanted to look at calcium signaling. And the reason is because if you remember to the first slide, the breakdown of PIP2 leads to a large increase in cytosolic calcium. And so what I did was I stained cells with a fluorescent calcium indicator, and then I added a PLC-gamma2 activator. And what you can see in this calcium trace here, where the common kind of variant, the P522 is shown in blue, the protective R522 variant is shown in red, and the PLC-gamma2 knockout is shown in yellow. When you add the activator, you get a bigger increase in cytosolic calcium within the R522 in comparison to the P522, which suggests increased PLC-gamma2 activity within this line and confirms observations in COS cells, which is good. And then within the knockout, we only see a very tiny increase, which suggests that our PLC-gamma2 activator is fairly specific. Okay. So having characterized this change in calcium release and activation, I next wanted to see whether or not it resulted in any functional changes within our microglia. So one of the things that I looked at was uptake of amyloid, and the way that I did this is I fed cells with FITC conjugated amyloid oligomers, and then what happens is these are taken up by the microglia and they fluoresce in green. And you can kind of compare the level of green within the cells as an indicator of phagocytic uptake. And here again you've got the common variant in blue, protective in red, knockout in yellow. And what we saw is increased uptake of these amyloid oligomers within both the protective variant and the PLC gamma 2 knockout. So because soluble amyloid beta oligomers can be toxic, perhaps one of the ways that the R522 reduces Alzheimer's risk could be by taking up some of these oligomers and reducing toxicity. Also, papers like this suggest that potentially microglial uptake of soluble amyloid oligomers could prevent downstream plaque formation. So one of the next things that we did is that Tom in the mice, he wanted to look in the mouse brain at kind of plaque size. And so in these graphs here, you've got the APP knock-in crossed with the common P522 in blue, and the knock-in crossed with the R522 protective variant in red. And you can see that within the cortex, we get a reduction in plaque area within the protective R522 variant crossed with the APP knock-in. And this is shown in these representative images here. And maybe this is due to the increased soluble amyloid uptake that I mentioned earlier. Okay, so we know that TREM2 is involved in amyloid uptake, and also that TREM2 is upstream of PLC gamma 2. So the next thing I looked at was TREM2 activation within these lines. And here, so again, blue, common, red, protective. And what I did is I stained cells with a fluorescent calcium indicator, and then I activated TREM2 using either TREM2 activating antibodies or DOPS liposomes. And what you can see is that within the protective variant, we have increased calcium release when activating TREM2, which suggests maybe we have increased TREM2 activity within these lines. Several papers suggest that TREM2 stimulation and activation may be dependent on PLC gamma 2 downstream, so this makes sense maybe in some way, hopefully. So we know that TREM2 overexpression can protect against some Alzheimer's disease phenotypes. So the hypothesis is that maybe the way that TREM2 overexpression is protecting acts in the same way as the way that the R522 variant is protective. Okay, so having looked at this, I next wanted to see if there are any other functional changes in the microglia. So this work was done with Hannah, who's over there. And basically, so this is the results from a facts-based assay which allows us to measure the concentration of different cytokines within cell media. And we looked at these 5 cytokines. And in this graph here, again, common blue, protective red, knockout yellow. And so the block colour bars, so the ones on the left, are ones that are untreated. And then the dashed bars are where the cells have been treated prior to the assay for 18 hours with LPS. And what you can see is that in the common variant in blue, we get an increase in IL-6, IL-10, and TNF following LPS treatment, but we don't see this increase within the PLC gamma 2 knockout, and we only see a slight increase in IL-6 within the protective R522 variant. So cytokines are very important. They regulate numerous functions, including cell growth, differentiation, and survival. And we also know that inflammatory cytokines are increased in Alzheimer's disease, obviously. And so this data suggests that maybe one of the ways that this R522 is protective is by reducing inflammatory cytokine release in response to activation. Okay, so having seen a couple of functional changes, we next wanted to try and kind of delve deeper into what might be accounting for these functional changes within the cells, and also to try and address a bit of a question, which is why in this data and in the amyloid data and in some other data, we got the PLC gamma 2 knockout seems to be acting in a similar way to the protective variant, even though we think that the variant is hyperfunctional. So this was done by Tom, and what we did is we looked at concentrations of PIP2 in the cells, and if you can remember from the first slide, PIP2 is the substrate of PLC gamma 2. And so what we did is we looked at PIP2 concentrations over time following PLC gamma-2 activation. And what you can see is that within the protective variant in red, when you activate PLC gamma-2, you get a much greater drop in substrate in comparison with the common variant. Now this kind of makes some sense, I think, because you've got hyperactivation of the enzyme, so this leads to more loss of substrate. But you can also see that it seems to take longer to recover to baseline in comparison to with the common variant. And this seems to be reflected within the mouse brain, whereby within the microglia, within the cortex and the hippocampus, in the protective variant, we just have a generalized kind of reduction in PIP2 concentration. So, as I said, we think the increased PIP2 degradation is due to the hyperfunction, and we think that maybe the reduced PIP2 could be preventing chronic activation of the PLC gamma-2 within cells, and maybe this could be what's leading to the overall reduced cytokine secretion, maybe. And it could also explain maybe why the knockout and the R522 have overlapping phenotypes, because even though you've got functional hyperfunction of the PLC gamma-2 enzyme in the R522, because this leads to chronic substrate depletion, you actually end up with maybe less function overall. So to conclude, the R522 variant has increased PLC gamma-2 and TREM2 activity, increased uptake of amyloid oligomers, which could be leading to decreased plaque burden within the brain, reduced cytokine response following LPS stimulation, and reduced PIP2, and the PLC gamma-2 knockout appears to mimic amyloid and cytokine responses of the knockout. And just so final slide. So what implications do I think this has for Alzheimer's disease? So because the increased enzymatic activity within the R522, this suggests that maybe activating PLC gamma-2 could act as a therapeutic target, maybe. And we also think the hyperfunction and substrate depletion might protect against AD by preventing chronic enzyme activation, which actually means maybe we want to inhibit the activity rather than activate it as a therapeutic target. It's kind of hard to know, really. And we think the increased uptake of amyloid oligomers is maybe leading to decreased plaque burden within the brain, and that the reduced cytokine response in the R522 could be helping to prevent the neuroinflammation within the Alzheimer's brain. Okay, thank you for listening. Well, that's fine.
Thanks, Emily. That was a brilliant talk.
Hi mate, yeah, thanks, that was really interesting. This might be a little bit unfair for you and might have to go to your chair, but why do you think that you see an effect in the plaque burden in the cortex but not in the hippocampus when you had really nice equal effect on, I think it was the PIP2 depletion across cortex and hippocampus? Is that right?
Yeah, that is.
Right. Yeah. So why do you think that you have like a really clear plaque effect in the cortex?
That is a really good question, but I maybe could have given the chat.
I wasn't told there'd be questions.
You're supposed to be asking them.
Well, yeah, but mine are really mean. So. Yeah, but basically, this work's sort of just finishing up now. We do see differences in the microglia response in the hippocampus and the cortex. The plaque percentage area response we saw there was one of the large ones. There were also changes in the number of plaques that we didn't show in this talk, and the size of some of the plaques as well. So the variation was quite high, and the actual significant effects were not enormous when it came to that, but that's quite a nice one there. It did seem to vary a little bit between cortex and hippocampus, male and female, and that was the 9-month one, I think. Yeah. So at 6 months they're a bit more even, and at 9 months seems to take off a little bit in the cortex. And that's as far as we've looked so far on these mice, so maybe at 12 months it'll catch up. The answer is I don't really know exactly why. I suspect there's subtle changes between cortical and hippocampal microglia and the way the plaque's being made now.
Cool, thanks.
Thank you.
Good answer.
Okay.
This might be a silly question, but why does low PIP2 reduce cytokines but doesn't affect the other functions? So it increases phagocytosis but reduces cytokines.
Well, you know, the funny thing is that we actually, so maybe I accidentally said phagocytosis, but the amyloid uptake is actually via endocytosis, but we actually see reduced phagocytosis of lysosomes and E. coli. So we don't really know how it's working. The PIP2 theory is kind of an early days theory where we're just trying to work out what's happening in this complicated model. When it comes to phagocytosis, the PIP2 is quite heavily involved in phagocytosis, which could lead to reduced phagocytosis, but it's not so much involved in kind of endocytosis as it would be with the amyloid oligomers, so that could explain why it's having less.
I also had one more question if I've got time. So this is more about the calcium stuff. So Adam Dinesh in Hungary has made these dread microglia. What do you think that causes? It just basically means that there's more calcium being released inside the cell, but what will it do to the cell?
Really? I mean, to me that seems, I don't know, like they'd just be super activated and everything would just be...
Yeah, so he's calling them activated, but what does it mean?
Well, I guess, increased calcium in microglia could lead to increased phagocytosis. Could mean that they're just eating more stuff up in the brain more generally. It could also, you could expect that maybe it would lead to increased cytokine release, because the increased cytosolic calcium can lead to more cytokine release in general. It could, I guess, even though we don't think that nitric oxide is released from microglia is a big thing in humans, it could induce that as well.
So
everything, yeah, pretty much.
Hi, fantastic talk. Have you checked the levels of TLRs across your PLC knockout and other variants?
No.
Because they're less responsive for LPS.
I guess we have not, so we haven't, but I think that it sort of is on the cards to look at
So more or less there will be comparable expression.
I guess we haven't really.
So recently we did some proteomics work. I think we'll have that, but but that data is still on my computer, like 6,000.
All right, sorry. Next time.
Okay, one more question.
Just had a question about your methodology. How did you detect PIP2 levels in time?
So we used several different ways of doing it. One thing was mass ELISAs. We also used some, that which I think is what that data was.
So
mass spec as well.
In the actual mouse, we mostly did fluorescence antibodies that were PIP2, and then combined that with IVA staining to get the microglia. But yeah, we used mass ELISAs for the in vitro stuff and also some antibody tests as well. We did a little bit of mass spec, but it was a bit difficult to get non-variable results. Okay, I think we're coming up to the end of the time for this. So, okay, we have Irish next.
That's not Irish.
I know. You're definitely going to deserve a drink.
Thank you, no worries.
So we now have our talk.
Guys, shut up. Okay, so hi, I'm Eilish. I'm a final year PhD student in Owen Peters' lab at Cardiff. So I'm keeping very much on topic with the talk of PLC gamma 2 here, nicely introduced there by Emily. I'm going to share with you some of my work from my PhD on the fly ortholog of PLC gamma 2 called small wing. And its glial role in modifying amyloid beta associated phenotypes. So for the most part of my PhD, I've been using Drosophila models to understand the glial roles of Alzheimer's disease risk genes throughout age. So for the non-fly biologists amongst us, you may be wondering how and why we can use Drosophila to understand Alzheimer's disease. So flies provide a simple in vivo system that allows us to study glial functions in Alzheimer's risk genes. So glial cells in the Drosophila brain make up around 10% of the total cell brain population, which is far fewer than that we know in mammals. However, they are well defined histologically and are amenable to powerful genetic analyses. So here's just a simple illustration outlining the various glial subtypes that we see in the Drosophila fly brain. So the glial cells in the fly brain do share functional and anatomical features with that of their vertebrate counterparts, in that their main roles are to provide trophic support for neuronal development and sculpting of synaptic connections. These Ensheathing glia subtypes also take on a role of engulfing degenerating axons, which are similar to that of the immune-associated microglia that we find in mammals. So I must point out that flies do not have microglia per se. However, it is the ensheathing glia that take on the similar phagocytic properties of that of microglia. Also importantly, we know that many of the Alzheimer's risk genes that have been identified from GWAS studies are well conserved in the fly, shown by the level of conservation in the DIOP score that we have here on the right. The DIOP score is just a score for conservation out of 15. And finally, there are many tools available in the fly that we can use to dissect glial functions of risk genes. So one that I've used throughout this project is the GAL8T, GAL4 temperature-sensitive binary system, which allows us to manipulate gene expression through temperature. It allows us to manipulate gene expression through temperature. So, for instance, we can use a glial-specific driver, such as repo, to allow us to express our gene of interest in a cell, in glial cells specifically. So the gene of interest we're very much interested in is PLC gamma 2. So this is because there was a rare coding variant identified from a 2017 GWAS study that showed there was a reduced risk of developing late onset Alzheimer's disease. So we know that PLC gamma is predominantly expressed in the microglia cells of the brain, and the enzyme is involved in catalyzing PIP2 into the secondary messengers IP3 and DAG, which are then involved in several signaling pathways that are to regulate cell survival, proliferation, and cytokine production. So studies have shown that the P522R protective variant confers a mild hypermorphic effect, which in turn increases the enzymatic activity of PLC gamma 2 and leads to enhanced survival, phagocytosis, and inflammatory response. So luckily, we can model the human PLC gamma 2 gene in flies with the fly gene called small wing. So flies only have one isoform of small wing gene, which there could be some functional redundancy. We know that small wing in flies is important for the eye and wing development, where we see in a knockdown or complete loss of the gene product of small wing, there is a notable reduction to wing size. And then from single cell expression data, we can see that small wing is rather evenly distributed in the fly brain across neurons and glia clusters. So the question that we wanted to address was whether glial small wing can modify amyloid beta associated phenotypes. So there are many models of Alzheimer's disease risk genes in the fly, but the one we've used for the following experiments is a secreted human amyloid beta 42 peptide that carries the Swedish Arctic mutation. So using the binary UASGL4 system that I've explained earlier, I've exclusively expressed the A-beta-42 peptide in glial cells. And as you can see, we see a marked, it's not very clear on here actually, but we can see a marked increase in amyloid beta accumulation, which was visible upon staining. So this is a control brain. It's really not coming out very well on here, but here's the control brain, and then the amyloid beta expressing brains. You can see the accumulation of amyloid beta. We also see a notable reduction to lifespan and locomotion deficits. So, however, what's really exciting is that upon glial knockdown of small wing, we rescue these amyloid beta phenotypes, in that we see an improvement to both survival and locomotor deficits by the 2-week time point. So we do see in both our control amyloid beta group and small wing knockdown group an accumulation of amyloid beta. However, from imaging, we cannot detect any drastic changes, and so we've moved to looking at using a more sensitive and quantifiable assay to detect the levels of amyloid beta load. So just to summarise that part of the work here, it's been a really interesting and novel finding to see that glial small wing can protect against these amyloid beta phenotypes. And that in itself is really interesting, but we want to now understand how this can happen. So one avenue that I've explored is looking at the changes of amyloid beta load. So we've used a really sensitive, highly sensitive and quantitative assay that allows us to detect and measure amyloid beta species in the Drosophila brain. So this assay is similar to an ELISA in that it coats the plate with capture antibody. However, it passes an electric current through the plate, causing the captured label to emit light. So from the assay, we've been able to quantify the amount of total soluble and insoluble amyloid beta protein within the fly brains. So as expected in our wild-type control group here, we do not detect any amyloid beta species as there is no amyloid beta being expressed in the fly. However, in our GFP control group, which expresses A-beta, we do see both soluble and insoluble forms of amyloid beta 42. However, the difference in amyloid beta detected in our small wing knockdown group is not significantly different from our GFP control groups. So this sort of suggests that the loss of small wing in glia is having a protective effect without changing the overall amyloid beta load. So another avenue we have explored is the regulation of PIP2 turnover. So the phosphoinositol pathway, being an important biological pathway, is well conserved in PIP2. Is well conserved in the flies. So both PIP2 and PIP3 do exist in the cell membranes of flies. So we know that PLC gamma 2 is directly involved in the PIP2 breakdown, which in turn subsequently impacts the PIP3 metabolism. So we know that we're therefore interested in using these lipids as a readout of PLC gamma 2 function. So what's really so PIP2s we know that PIP2 and PIP3 play important roles in plasma membrane functions such as phagocytosis and regulation of actin cytoskeleton. What's really interesting is that we can use these PIP reporters in flies, so we can actually monitor the dynamics, the membrane dynamics of these lipids in vivo. So here's just an image of the PIP reporters that we have, the PIP2 reporter that we have in the fly brain. So these reporters are a GFP tagged protein tagged to the plextrin homology domain, that can recognise the head groups of PIP2. So here in the green you can see GFP, which is labelling where PIP2 is in the glial cells. That's just a bit closer up. So what we've then had a look at is what knockdown of small wing looks like in our PIP2 models. So knockdown of small wing looks, which we expect, looks to result in an accumulation of PIP2 levels. So we were expecting to sort of see this increase in PIP2. However, from this image we sort of see that there is not only a change in increase of PIP2 levels, but also a varied sort of distribution of PIP2 as well. It's not uniform across the entire brain. So finally, I just want to go over some of the later work that I've been working on, and that's modelling the protective P522R variant in the fly. So I've made transgenic fly lines that carry the wild type and protective variant. We've then gone and looked at whether the human PLC-gamma2 gene can rescue effects of loss of small wing. So previously I spoke about that loss of small wing causes a wing phenotype, reduced wing size, and when we introduce the human PLC-gamma2 gene, this does in fact rescue the wing size, as shown here. So this sort of suggests that the human PLC-gamma2 gene, small wing, are functionally conserved. So then finally we want to look at whether the AD-associated coding variant can alter the amyloid beta and PIP2 phenotypes, such as in survival, locomotion, amyloid beta load, and levels of PIP2. So just to main take home from this talk is that I want to tell you that loss of glial small wing we see rescues amyloid beta associated phenotypes in flies. We don't see any, this occurs by, we do not see any changes in amyloid beta load. However, we do see a change in PIP2 membrane dynamics, which is affecting both the levels and distribution of PIP2. We would like to look at where amyloid beta may localize in relation to this PIP2. So does amyloid beta co-localize with in areas of increased PIP2 accumulation? As well, we would like to understand whether amyloid beta is whether loss of small wing is impacting the amyloid beta toxicity. So we would look at markers of cell death, autophagy, or ubiquitinated proteins. And finally, we are modelling the human PLC-gamma2 wild type and protective variants in flies, and we'd like to understand whether this protective variant does alter amyloid beta neuronal phenotypes. And with that, I'd like to thank you for listening, and I'll take any questions.
Thanks, Alicia. That was really great. I have a question, first off. With the small wing, when you had the knockdown and you saw the accumulation of PIP2 in different areas, what kind of areas were those?
So the reporters exclusively expressed in glial cells, so the reporter should be in all glia. I don't know specific like glia subtypes that it is in the fly, but we're trying to understand where this PIP2 is being localized to. At the moment, I don't know yet.
Because that'd be really interesting, especially what the P522
Yeah.
Yeah, thanks, great talk. I really like the A-beta model, is really, really cool. I couldn't quite tell from the pictures, but so you obviously get insoluble and soluble A-beta.
Yeah.
But does it form like plaques? And then if it does, do you get, I know you don't have microglia, but your versions of microglia around plaques, and do you get dense core plaques and things like that?
It's still very hard to tell that with the fly model.
Yeah.
I feel like when we're looking at it in the fly brain, I wouldn't say plaques per se, but then obviously I've used this MSD assay where I've done insoluble protein and it's detected it, so I guess I can say there is
yeah, they might be forming like fibrils. Yeah. That's really cool. I guess, like, to what extent that's modelling the sort of plaque that we get, you know, in humans and mice would be really interesting to see what the like immune response to your various different species of amyloid in the fly brain are. That'd be really cool.
Yeah.
Following up on that, do you know whether there's any cell death associated with the plaques? Because the mouse models of amyloidopathies are so the cells, the amyloid accumulates, but the neurons don't really die very much.
Okay.
If at all. So if you have that capacity to model it in the fly, that's super cool.
Yeah, I mean, we're wanting to look at cell markers, markers of cell death within the fly upon these amyloid beta models. At the moment, I've looked at a couple of markers of autophagy, but not yet cell death, so we'll see.
It'd be cool to look at synapse loss as well.
Yeah.
Cool.
I guess, could you use the ring assay as like a means to know whether there's like cell death and neuron death going on?
I feel like it'd be a very crude, like, sort of readout, more to see, like, you wouldn't really know, like, what type of cell death you're getting, I guess, from just doing the locomotion, like, assay.
But there's quite a strong phenotype, isn't there?
Yeah, if there's a, well, I think the phenotype for ring wasn't super strong. It wasn't, like, super, it wasn't, like, a really strong locomotion deficit. But I think you're more likely to see what's going on inside the brain by doing, like, the immunostaining and stuff like that. Yeah.
You know, you looked at amyloid 42.
Yeah.
How about the 40-42 ratio?
So in the MSD assay that we used, we were able to actually look at different species. So the A-beta 38 and 40 in the fly, I didn't actually detect any A-beta 40, but I think that's because flies obviously don't make amyloid beta themselves, and we're just expressing a human peptide, so we're only expressing the 42 version. So it's kind of, that was like nice in the way that our model's working. We're only like producing the A-beta 42. So yeah, we don't get that ratio.
Okay. Thank you very much.
Thank you.
Okay, next up, I think we have David. That's what it says.
I think so.
Yeah.
Great, so David.
Okay, yeah, hello, I'm David Munro. I'm in the Priller group at the University of Edinburgh, and I'm going to talk generally about CNS macrophages and try and address this question of what happens to the brain when they're not there. This is, which one is it? Oh no, he's in it, sorry. So you'll all be aware that the CNS is compartmentalized. You have the parenchyma of the brain, and you also have lots of extraparenchymal or border compartments, so such as like the meninges and the cerebroventricles and the choroid plexus. And something that all these compartments have in common is that they all host macrophages. Studies have kind of nicely shown recently that the identities of macrophages in the CNS are very like compartment specific. Obviously you have the microglial signature that everybody talks about, and you can see here that the macrophages in the choroid plexus and subdura and the dura, they're all kind of distinct from each other. So these are all known as border associated macrophages. Despite their differences, they do have similarities. So they have this kind of core BAM signature where they have high expression of things like ApoE and MS4A7. We also have this little population over here of choroid plexus associated macrophages, which are actually sitting on the surface of the choroid plexus. They're exposed to cerebrospinal fluid, and unlike the other BAMs, they don't express these core BAM signature genes, and instead their identities are a bit more microglia-like. So what happens to the brain without microglia? Well, there's this kind of emerging concept that it might be better to have none than to have bad ones. So that's suggested by these papers, for example. But a lot of these claims are based on rodent studies. So obviously when microglia are absent in humans, they don't fare too well. And depletion in these studies is often done over like a kind of defined period of time rather than them being absent from the start of development. There's also this potential issue where you're depleting like 10% of the brain cells, you're left with the corpses and you're getting rid of the cells you would want there to clear them up. So that could influence the environment as well. So I'll be talking a lot about these mice, the CSF1R fire mice, which Veronica mentioned about briefly. They completely lack microglia. You can't see this very well, but they're completely gone in the brain, whereas some border-associated macrophage populations are retained. So this is a knockout here. And basically, yeah, we wanted to understand, are these mice going to be useful in kind of getting over these issues related to the importance of microglia during development, for example. So we wanted to know, are they really absent from the start of development, or is it that they're kind of arriving during development and then they're absent in the adult? And to address this, we looked at embryos at the stage where macrophages start arriving in the head, and we kind of divided the brain up into compartments, the cephalic mesenchyme, where the kind of primitive BAMs arrive, the CNS parenchyma, where the primitive microglia will arrive, and the cerebroventricles, where macrophages also come in. And you can see so they start arriving about E9.5, and then they kind of increase in numbers exponentially over the ensuing days in these control embryos. But if you look in the fire mice, there is a little bit of an effect. The BAMs are arriving, but it's kind of blunted compared to control embryos. Then if you look at the CNS, so the primitive microglia, again you're getting an exponential increase, although they arrive slightly later than the primitive BAMs. And if you look at the knockouts, the arrival of these cells are just completely gone. And this is also true for those that are coming into the cerebrospinal fluid system. And just to kind of characterize this a little bit further, it's been shown that there's this early developmental divergence of macrophages arriving in the head. So already the primitive microglia are different from these BAMs, and that can be marked by, for example, CD206 expression. So you can see both populations are present in the kind of control embryos. And even at this early stage, it's recently been shown that these differ in their kind of dependency on certain cytokines. So primitive microglia are TGF-beta signaling dependent, whereas the BAMs aren't. And if we look at the fire embryos, it's really just the CD206 positive border-associated macrophages that are coming in, and nothing in the parenchyma. So just to summarize, that was embryonic, but it's the same is true in the adult. So the microglia are completely gone throughout life, and it's not completely specific. So all the different macrophage populations in the cerebrospinal fluid are also absent, which is characterized a bit further in there's a paper. These populations, which I mentioned, have that core BAM signature. They're all persisting in these mice, but they're not retained to the exact same levels, at least not in the case for these stromal choroid plexus macrophages. And we want to understand, are they able to persist because they don't rely on the fire element for their expression of CSF1R, or are they actually really low in CSF1R levels but able to persist in the absence of the high levels of the receptor? So to try and understand this, we did some flow cytometry for CSF1R, which is also known as CD115. So the dots here are labeled based on their kind of intensity of expression for CSF1R. And obviously, as you'd expect in the wild-type mice, the microglia and other myeloid cells are super high for CSF1R levels. Interestingly, the heterozygous mice have reduced levels. So they've got approximately half the amount of CSF1R in both cell types, suggesting it's kind of a useful model to explore CSF1R haploinsufficiency. And in the knockouts, of course, the microglia are absent. And if you look at the myeloid cells, so this is quantified over here, they are affected, so their CSF1R levels are very low. So it seems like they're just less dependent on it. But if you look at their full transcriptomes, they're not super affected, so it seems like they should be functional. So to summarize this model, these mice survive well into old age. Unlike if you have a CSF1R full knockout mouse, they don't. They have more global depletion of macrophages as well, compared to this line. It's a good model to explore the importance of microglia during development, since they never arrive, and this point of them never arriving makes them a good model to explore the consequences of their loss in the parenchymal environment, because you don't have that issue of lots of cell corpses potentially influencing the cells in an unrealistic way. But it's important to note that it's not particularly, the impacts aren't limited only to microglia, so in other organs and BAMs are slightly affected too. So just bear that in mind when I go on to talk about the effects. So this is I'll start by talking about behaviour. We did a few tests. Started off fairly basic with an open field test. These are just some representative examples. The knockout mice is going to be over here. Oh, sorry. So yeah, this guy obviously doesn't have any microglia, but you can see him running about fairly normally compared to, you can't really tell by eye any differences between the wild type and the heterozygous counterparts. And if we quantify this, so that's the open field, you can look at their locomotor behaviour, and we're not finding any significant differences in the amount they're moving about. So the knockouts are shown in red here, and the females and males. And also, we're not finding differences in where they're placing themselves within the maze. So mice prefer to stay around the edges, and that's what they're doing in the knockouts and in the other mice. We then did a elevated plus maze test, which is a more kind of a measurement of anxiety. So you've got these closed arms where the mice prefer to be because they feel safer, and then they'll occasionally go out into these open arms where if they're feeling brave. And you can see this is a knockout here, kind of moving about in the same kind of way as their litter mates. And quantification of this shows that the amount they're travelling doesn't differ between the genotypes, and they're not spending any more or any less time in the open arms than the other genotype mice, suggesting there's no kind of baseline anxiety in these mice. We then did a novel object recognition test with the familiar object being a Lego kind of tower and the novel one being a light bulb. So all groups are showing preference for the novel object, as you'd expect, and you can tell that by the means being over this dashed line, but you can't see any differences between the groups, so it suggests there's no differences in recognition memory. Then we wanted to understand, are the identities of kind of the cells around microglia impacted by their loss? Like, is their identity dependent on microglia? So there's this idea that cells, your cellular neighbours, especially for microglia, it's thought that, for example, neurons and astrocytes will regulate the phenotypes of microglia. And we want to understand, is there reciprocal signalling from microglia to regulate the identities of other cell types? And to do this, we compared these wild-type mice where microglia are present versus the knockout mice where they're absent. And we did this with mice that were 1 year of age. We took a hemisphere from the brain and dissociated it and tried to get a really good kind of spread of different cell types to analyse. I should mention this paper from the Rubin lab. Methodologies was quite helpful in kind of giving me some advice about the protocol he used. So we adjusted his protocol and got quite a good spread of different cell types, lots of astrocytes, oligodendrocytes, endothelia, and obviously microglia. But I should mention that neurons are quite underrepresented using this protocol. So if you're to compare the kind of spread of cells in the wild types versus the knockouts, obviously in wild types you have all the cell types are present, whereas where you compare the knockouts, thankfully, the microglia were absent, which is good. And if you look at the other cell types, there's no kind of obvious kind of missing cell or shift in clusters. And to kind of examine, are there a lot of gene expression changes in the different cell types when microglia are absent? We did it more statistically by doing actual differential expression analyses. And you can see that there are actually surprisingly few changes, suggesting that the transcriptomes of most brain cell types are relatively maintained when microglia are absent. But we're seeing relatively more changes in the oligodendrocytes. So oligodendrocytes, in the same way that when you have a diseased brain, microglia can become activated, this also can happen to oligodendrocytes, and lots of studies have recently addressed this and found that, so MS and AD models have found that there are kind of a few genes are really changing consistently in disease states, and two of those are C4b and serpin A3N. And in these fire mice, when we compare them looking at the oligodendrocytes, you can see there's this upregulation of C4b, and in serpin A3N and the knockout, suggesting that in the absence of microglia in 1-year-old mice, the oligodendrocytes are becoming reactive. And we're going to follow up on this. So just to conclude all of this, macrophages are differentially dependent in the CNS on the fire element of CSF1R. Brain development seems to proceed without obvious defects in the absence of microglia, and the baseline behaviours that we examined at least didn't significantly differ when microglia were absent, and neither did the transcriptomes of most brain cell types. So I think collectively this suggests that, I mean, obviously microglia are important for various things, but perhaps like you could read a review and come out with the impression that things are really going to be messed up when microglia are absent, but it doesn't really seem to be the case, so maybe people overstate their importance in certain processes. And thank you to all these people. I wasn't expecting that to be controversial. My boss, Josef, thanks to him. Nadine did a lot of the bioinformatics stuff from Anna's lab. Miron, a lot of the behaviour. Steph, over there, helped a lot with the protocol development and the brain dissociation, so thank you, Steph. From Barry's lab, Claire generated the mice initially, Samantha and Chris, who are like developmental hematopoiesis experts, and Barry Bradford and David Hampton, who helped with like tissue processing and things like that. And thank you all, and thanks to the organisers for setting it up and the opportunity to speak. Yeah.
That's a really interesting model. I was curious, have you tried crossing this model with any disease models to see what happens there?
I haven't personally, but people have done it, and I think there'll be papers coming, I hear.
Okay.
Yeah. I can't say much because I've not seen the data, but I know it has been done with for some models, yeah.
The Prion model that did it with...
Oh, sorry, yeah, sorry.
It's published now.
Well, yeah, that's not with another transgenic line though, is it? It's like they just give them prior infection.
But it's a disease model.
But yeah, sorry, yeah.
Genetic cross. But they're worse in prion disease. The fires.
Worse than ours.
Yeah.
I mean, sorry, I always ask you.
No, you can put it first.
Okay, in the Claire's initial paper with the fires being characterised, they do an experiment like, okay, what is like overall phagocytic capacity in the brain? And they show there's no difference, even though there's no microglia, and they kind of imply, oh, probably other cell types have taken on that role. You would expect, if like suddenly astrocytes are becoming really phagocytic, you would definitely expect to see that by single cell.
Yeah.
So do you think why then, what's taken on the role of the phagocytosis in the brain basically, if no other cell types are changing?
Yeah. Yeah, we kind of thought maybe we were going to see astrocytes upregulate phagocytic genes, but we didn't. And I don't really know what would be compensating because we don't have massive shifts in terms of like we're becoming pro-phagocytic. So from any cell type that we looked at, so I don't know. But it's a good question.
Cool.
Hey, nice talk. I think it's really intriguing difference between this model, the fire model, and the full receptor knockout, so the OPOP mouse model. And I was wondering if you can comment a bit more on that. So I think the only difference there, an important difference, as you mentioned, is that with the full receptor knockout, there is also peripheral macrophage deficiency, whereas in the fire, this is less so. So could it be that the reason why you don't see that much of a phenotype in the fire could be the result of the peripheral macrophages not being affected, and they might be contributing then to the phenotype that you see in the full receptor knockout?
Yeah, so basically there are actually quite a few macrophage populations that are peripheral ones that are lost in the fire mice, like the kidney macrophages, for example, are gone. But obviously with the full knockout, it's more global, and it's speculative, but I mean, perhaps there's certain populations in that are missing in the full knockout, but not the fire, that are particularly vital, but nobody really knows yet, I'm afraid, so can't really give you a good answer.
And what do you think what happens with the empty space when the microglia are not there?
Well, it's not going to be empty because other cells will kind of like
other cells will
be in those places. Yeah. I mean, I think it just suggests that microglia really are just immune cells and they're to like act as like watchmen and make sure things are going right. And they become particularly important in disease settings, obviously. Yeah, I'm not too sure.
we've got some questions on the chat. So Colin Crawford, who's a new postdoc with us in Barzop, has said, do you think the changes in oligodendrocyte gene expression is related to regulation of myelination? Are they normally myelinated at the level of G ratios, internode length?
I think this is more of a question for Neve and Veronica, to be honest, because they know a lot about this stuff. I think you'll be able to get a good answer soon. Honestly, I don't think I'm the person to answer that very well.
Do you want to say anything?
Come see my poster.
Come see.
The myelin's not normal, so.
Okay.
Yeah.
Not normal myelin, Colin. And then Jamie Lone has asked, I understand that loss of fire is associated with hydrocephalus in a background-dependent fashion. Does this data give any insight into why?
My data doesn't really give any insight, no. The idea is that, so I think the first couple of litters of fire mice were all, when they were knockouts, were all developing hydrocephalus. Hydrocephalus is a very common phenotype in humans that lose microglia as well. And other mouse models where microglia are affected, they get hydrocephalus, or at least in large ventricles. But yeah, the kind of background-dependent differences in the fire mice aren't actually proven. I think it was claimed, but I think there's now kind of a lot of evidence that that's not really the case, because other people have the background which is supposed to get hydrocephalus, and those mice are fine. So, yeah. Hello.
Have you got any idea what C4B might be doing?
It's a complement subunit, so.
Yeah. Mostly just, hi, we work at a complement lab.
Again, I don't think I'm going to be able to give you a very good answer. Like, this is fairly new data, and it's not something I've personally investigated very much. So.
Cool. I mean, we work in a complement lab. We wouldn't go so far as to call ourselves complement experts, but feel free to say hi in a coffee break.
Cheers.
David, I'm not a single cell expert, I've never done, but just a curiosity-driven question, that why do you not find neuronal signature in your data? Do they die or processing?
So,
I don't know.
Yeah, the original paper that I was sent from the Rubin lab, they didn't sort, so they had really messy preps with lots of brain kind of debris and stuff they didn't clear out, and they didn't sort, but they just put so much money into it to get like lots of neurons, but we basically sorted out the debris to get a better return of cells at a less expensive price. So yeah, it's basically the sorting process, I think. Not killing the neurons, but maybe we were just avoiding them with our kind of selection of cells. Or it could have been both. Like, obviously neurons are pretty sensitive, so they don't like being sorted. We tried to sort very slow with a wide nozzle and all that, but yeah, we hoped to see neurons and we just didn't.
Have you looked at the effect of inflammatory stressors on the behaviour of these mice or the lifespan?
I haven't. I think those experiments are also being done. I think there's a lot to come quite soon. Hopefully you'll find out soon. So sorry, I can't tell you much.
Okay, great. For the sake of time,
cheers.
Thank you. Okay, so next up is Paige.
Laser pointer, was it?
Yeah, just the red bit.
Ah, gotcha. Okay, great.
Thank
you. Hi, so my name's Paige Mumford, and I'm a PhD student and research assistant at the UCL DRI in Dr. Francis Wiseman's lab. And after that talk about how microglia aren't important, my talk so we'll see. You should have gone last. Okay, yeah. So I'll be speaking to you about my PhD project, which is on preclinical modeling in the mouse of altered neuroinflammation in Alzheimer's disease Down syndrome. So we're a Down syndrome lab. Okay, great.
So I'm assuming nobody else here researches Down syndrome, so I'll just give you a brief detail of that. So people with Down syndrome have 3 copies of chromosome 21, which encodes over 200 protein coding genes. And having 3 copies of this chromosome leads to a range of phenotypes that affect multiple organ systems throughout the body. Probably the most well-known characteristics of Down syndrome include intellectual disability, congenital heart defects, high susceptibility to infections, and for everyone in this audience, early onset Alzheimer's disease. So why do people with Down syndrome get Alzheimer's disease? The gene for amyloid precursor protein, or APP, is located on chromosome 21. So people with Down syndrome have 3 copies of APP, and this is the genetic cause for such a high prevalence of early onset Alzheimer's disease in people with Down syndrome. So universally, all people with Down syndrome will develop A-beta plaques, so one of the hallmarks of Alzheimer's disease pathology, by the age of 40. And if they live long enough, will eventually develop the other pathologies and dementia. So there's this clear link between 3 copies of APP and this prevalence of AD and Down syndrome, but our lab is interested in the role of the other chromosome 21 genes. So how does having 3 copies of all these other genes modify the development of AD in people with DS? And multiple other chromosome 21 genes have been implicated in different mechanisms known to be involved in AD. So it would make sense that having 3 copies of these genes would be modifying the disease development in this population. So we'll be focusing on, in our lab, we're focusing on how neuroinflammation, as we all know, an important aspect of AD, is changed due to having these other chromosome 21 genes in 3 copies. So what's known about neuroinflammation in ADDS already? So it's been shown in a few papers very recently published that Down syndrome brain has an altered microglial morphology compared to euploid controls, as well as changes in pro-inflammatory cytokines in the brain across ages, both preceding AD pathology and as AD pathology progresses. And there's a distinct neuroinflammatory profile in ADDS brain compared to sporadic AD brain. So there's definitely something changed in neuroinflammation in microglia in Down syndrome, but what chromosome 21 genes are responsible for this is unknown. So I'll also mention it's known that the peripheral immune system in Down syndrome is also very generally perturbed. So this includes elevated pro-inflammatory cytokines starting at a very early age, and then also this hypersensitivity to interferons, which are part of the antiviral response. So essentially, people with Down syndrome are constantly fighting a viral infection that doesn't, even if it doesn't exist. And there are multiple chromosome 21 genes that have been implicated in such immune system differences in Down syndrome. And for my project, I'm just going to focus on 5 of them, because there's over 200 genes I could be looking at. So the top 4 here are interferon receptor genes. So IFNAR1, IFNAR2, IFNGR2, and IL-10RB. These are all chromosome 21 genes that encode subcomponents for interferon receptors. So people with Down syndrome have 3 copies of these genes, and this is thought to be responsible for this interferon hypersensitivity that's been reported. A 5th gene that I'm also interested in and maybe modifying immune system in Down syndrome is RUNX1, which encodes a microglial transcription factor. But for the most part, I'm interested in these interferon receptor genes and how these particular genes might be altering neuroinflammation when found in 3 copies in people with Down syndrome. So to study these genes, we're using different segmental duplication mouse models of Down syndrome. So the genes of human chromosome 21 have orthologous genes spread out in the mouse genome across 3 chromosomes. And we use different mouse models which have 3 copies of different groups of these chromosome 21 orthologous genes. So for my project, I'm using these 2 models, the DP1-TIB and the DP2-TIB. Now the DP1-TIB has 3 copies of this very large region of chromosome 21 orthologous genes, so about 148 of these genes are in 3 copies in this model. Whereas the DP2-TIB model has 3 copies of a much smaller subregion, so only 33 genes. And both of these models have 3 copies of my candidate genes that I mentioned previously. So we can use models like these to essentially map phenotype to groups of genes that could be causing that phenotype. Okay, so using both of these mouse models, since I'm interested in their effect on neuroinflammation, my first step was to establish that my candidate genes of interest are in fact elevated in expression in the brain tissue of these animals. And that's exactly what I found in the DP1-TIB and DP2-TIB, all of my candidate genes were elevated in expression due to being in 3 copies. And that might seem straightforward, like you have 3 copies of the gene, of course you have elevated expression, but actually that's a huge thing that people are researching in Down syndrome. Just because you have 3 copies of a gene does not necessarily mean you have elevated expression of the gene. So this was an important thing for me to establish. And also, I wanted to make sure that both of my models were adequate models in which to assess the effect of these genes in 3 copies. But I'll mention my work on this question is not yet done, because I've only shown that in total cortex and hippocampus you have elevated expression of these genes. I'm looking specifically at microglia. What is the effect on microglia of having 3 copies of these genes? So my next step is to confirm that these genes are elevated in microglia specifically, because dosage sensitivity can differ by cell type. So that would be my next question there. So I'd shown that in the DP1-TIB and DP2-TIB models, all of these genes that I'm interested in are elevated. Now, how does that affect neuroinflammation? So I did just kind of general characterization of neuroinflammation. First, I quantified the levels of IBA1 positive microglia in the hippocampus. And in the DP1-TIB, which again has 3 copies of this very large group of genes, I found an increase in total microglial number in the dentate gyrus at 6 months of age. And this is in line with other groups who've also found increased microglial number in a similar mouse model, and also evidence of altered hippocampal microglial morphology. And this also is in line with our collaborators who found at 12 months of age that the same mouse model has increased microglia everywhere in the hippocampus. So maybe with age, this model in particular is having increased microglia in the hippocampus. So then I did some quantification of different pro-inflammatory cytokines, again, kind of general neuroinflammatory markers. And I found that consistently across ages and brain regions, I was seeing increased interleukin-1 beta levels in this mouse model. So this is a phenotype that's also been seen in people with Down syndrome, and relevant to the AD field, we know that interleukin-1 beta is produced by activation of the inflammasome pathway present in microglia, which is upregulated in AD. So we've got a PhD student, Cleona, sat over there, who has a poster number 4 that's specifically on this. So her project will be looking at the inflammasome pathway with this model. Next, since I'm interested in that interferon pathway, I next looked at the expression level of different interferon-stimulated genes, or ISGs, in the brain tissue of these mice, and all of these genes here are some of the genes which are upregulated in people with Down syndrome. So when I looked at the levels of these genes in the DP1-TIB brain, I found that OAS2 levels, but none of the others, were increased in expression in DP1. So this is maybe a little bit of evidence that there's an interferon signaling active in this mouse model, and this also is in line with other groups which have found similar things. I'll also mention, while I'm not specifically focusing on the complement cascade, I also found that 2 genes which are involved in the complement cascade were up in this mouse model, so C1qa and C3. And we have another PhD student in the lab, Rian Llewellyn, who I think will be in tomorrow, who will be focusing on this particular pathway, how is complement altered in Down syndrome. So I've found these phenotypes in the DP1-TIB, which has 3 copies of that, like 148 genes. Now I wanted to look at a group of smaller genes. So can I map any of those phenotypes to a smaller group of genes? So all of this data is in the DP2-TIB mouse, which has 3 copies of just 33 of those genes. And I didn't see any of the same phenotypes. So in the DP2, there's no change in dentate gyrus total hippocampal number. There's no change in interleukin-1 beta levels. But interestingly, there was an increase in interferon gamma. So this is maybe evidence of interferon signaling in this mouse model. And also, elevated interferon gamma has been found in tissues of other Down syndrome mouse models. And of relevance to microglia, interferon gamma stimulation to microglia by other groups has shown to induce a morphological and functional change. So maybe the microglia in this DP2-TIB model are functionally different due to this increase in interferon gamma and also having 3 copies of those receptors. But that has to be further investigated. So again, when I looked at the same ISGs, I wasn't able to replicate the DP1-TIB findings, so no change in OAS2, no change in C1qa or C3. I saw some mixed findings in IFI27, But overall, I think this is showing there's pretty little evidence that there's interferon hypersensitivity in this model, which was disappointing. But my next step will be to probe the system. So we think that maybe you need to give the system some kind of stimuli, see how it responds, and the response itself is hyperactive because of these extra copies of receptors. So that's my future work, focusing more on the interferon pathway in this mouse model. So I'll be using this ex vivo organotypic hippocampal slice culture method from DP2 tib mice, treating those cultures with interferon beta, which is a type 1 interferon, and then seeing if the microglia from the DP2 tib, which have 3 copies of these interferon receptors, are hypersensitive to interferon stimulation. And I would hypothesize that they are, but we'll see. And then bringing in the Alzheimer's part, because we're ultimately an Alzheimer's disease Down syndrome lab, I then want to look at if the microglial response to amyloid is altered by having these interferon receptor genes in 3 copies. So interferon signaling is known to be involved in the microglial response to amyloid. Elevated ISGs are seen in microglia surrounding plaques, and if you block interferon signaling in amyloid mouse models, you get a reduction in microglial-mediated neuroinflammation. So I'm interested in this interaction between amyloid and potentially interferon hypersensitivity. So I'll be crossing my DP2 tib mouse with the APP-NLGF knock-in mouse, which I'm sure other people in the group are working on, and then I'll compare the microglial phenotypes between these two. So just a model with amyloid, and then a model with amyloid and my interferon receptor genes in 3 copies. So that's the ultimate goal. So in conclusion, I found that the DP1 tib model, which again has 3 copies of a ton of genes, has this neuroinflammatory profile characterized by elevated microglia, elevated interleukin-1 beta, and OAS2, C1qa, and C3 expression. But these phenotypes were not present in the DP2. So this is suggesting what other genes could be causing these changes. There's about 112 genes which are in 3 copies in the DP1, but not in the DP2. Any of them could be causing it. That's a future student's problem. But I'll just suggest one gene that they could look at is USP25. So this gene, again, chromosome 21 gene, which is in 3 copies in the DP1, but not the DP2. This gene, a paper just came out showing that, so it's a deubiquitinating enzyme. And this paper that was published like 4 months ago found that if you overexpress USP25, you kind of increase this microglial-mediated neuroinflammation, and if you knock it out in the 5xFAD mouse model of amyloid, this reduces the inflammation. So potentially in the DP1 tib, you have 3 copies of this particular gene. Maybe that's contributing to neuroinflammation. Again, someone else's work. But my future focus will be on these interferon receptor genes, how having 3 copies of them, as you do in the DP2 tib, is altering the microglial response to A-beta. And just briefly, our hypothesized pathway for this. So we know that there are 3 copies in Down syndrome of these interferon receptors. This leads to an elevated interferon response. Others have shown that if you treat microglia with interferons, you get a microglial response. And A-beta plaques in AD also induce an interferon response in microglia. So taking all of these things into account, we think that in our mouse model cross that has both amyloid and the interferon receptor genes in 3 copies, we'll get a heightened neuroinflammatory response from the microglia in response to the amyloid. But that's the next step. So thank you so much for listening, and please let me know if you have any suggestions for the project as well. I've got 2 more years. Thank you so much.
Thanks, that's really interesting. I think the APP cross sounds really fascinating. How that model develops. I was wondering if you'd looked at the developmental changes at all, like silence pruning and stuff like that.
So yes, I've been asked about that. No. So the DP2 tib mouse model is not really used that much because so few genes are in 3 copies. I think some people have looked in the DP1 tib, so with the very large group of genes, and there are definitely neurodevelopmental changes in that model, which makes sense because there's almost 150 genes in 3 copies. But the DP2 tib specifically, not sure.
Fair enough.
Yeah, sorry.
Any questions?
In Alzheimer's disease patients, do they show white matter pathology at all?
Oh, that is a good question. I'm not totally sure. I've seen...
I'm sorry, I meant Down syndrome. Sorry.
No, I knew it. Alzheimer's disease, Down syndrome, yeah. I got it, don't worry. That is a great question. So I'm not actually sure. I don't think so. So the only things I've read about white matter in the ADDS brain has been microglial focused. And in the white matter, I think they do see changes in microglia there, but I don't know about, I'm assuming you're talking about like white matter hyperintensities or something like that.
Yeah.
I don't think so. Cleona? No. Pulling her in, sorry. I don't think so, I'm not quite sure. I'll look into that. Thank you.
Thanks very much, Pence.
Thank you so much.
It's like dictionary corner. It's like dictionary corner.
Yeah, cool.
Okay, so we have our last talk for this session. Karis from Co.
Sorry, there's my earring. I'll just put that there. I'll do it one earring. Hi, I'm Karis. I was going to make a joke about my clear not being important, but Paige got there before me. But I am currently a fourth year student, which makes me feel really ill, at Cardiff. I was part of the Hill lab, but he left, so I was adopted by the Phil Taylor lab. It's a little bit awkward. But I'm going to talk a little bit about my PhD project. More specifically, the proteomic investigation of MEF2C in microglia-like cells. So a little bit of background. Everyone will know that there are now over 50 susceptibility loci for Alzheimer's disease, and this implicates immune cells as probable factors of the risk. The gene that we're interested in is MEF2C, which codes for a transcription factor which regulates target gene expression in microglia. So a little bit more about the protein itself and why we think it might be important. So a recent study integrated GWAS results with sites of open chromatin and found that AD risk SNPs are actually enriched within particular microglia open chromatin regions that have MEF2C DNA binding motifs. So what they think is going on is that genetic variation at these risk SNPs means that MEF2C can't bind or can't regulate gene expression, which means it has an impaired transcriptional output. So this actually implicates MEF2C's transcriptional network as an AD genetic risk mechanism. So to look at this, so basically we want to look more into MEF2C and how it functions. So we want to understand how MEF2C is actually regulating this gene expression and therefore the AD risk, and also determine what's it working with, what regulates it, to see if we can find out more about what MEF2C is doing. So there's two main approaches to this. There's the proteomics, which we're looking at the interactome of MEF2C and by a method called co-IP mass spectrometry, and then also genomics. So MEF2C is actually a calcium signaling integrator, so we want to look at the effects of calcium signaling on the chromatin landscape using a TAC-seq, and hopefully, if I have time, the gene expression profile with an RNA-seq. But today, the work I have, which looks nice, is proteomics, so I'll talk a bit about that. So just a little bit about co-IP. I'm not sure many people do proteomic stuff. But basically, you can isolate an entire protein interactome from a cell with an antibody that's specific to your protein of interest. So basically, you lyse your cells, and it will have your protein of interest, any kind of interactors attached to it. You add in some antibody, which obviously binds to your protein of interest. Add in some magic magnetic beads, which are really cool, which just allow you to put on a magnet, and it basically brings out the entire complex. Purify that, look at it by Western blot. I've probably done about 70 of these now, so I've done a lot of those, and then it gets sent off for mass spec analysis by some really clever people. So it's just an example of what the co-IP should look like. So you obviously see some of the protein of interest, so MEF2C is about 50 kilodaltons. You should see a bit in the input, it's got to go in to come out. Then obviously most of your protein should be in the elution at the end of the IP because you've isolated it from your cells. We also run a control, an IgG, which means that it will capture anything that non-specifically binds to antibody, and we then compare those two lists and kind of use it as a background filter. this gets sent off for mass spectrometry, where basically they digest the protein into lots of little different peptides, and they can use those peptides to figure out what else is actually within the sample. So what mass spec can tell you is a little bit more about your protein of interest itself. So there were 29 peptides that identified MFGC within the set, and these span about 46% of the protein. We found 2 isoforms within that, so isoform 1 and isoform X3, and there were peptides that were specific to both of those, so we think it actually exists as 2 main isoforms in BB2s. I think I forgot to say the microglia-like cells are BB2s. But importantly, these isoforms actually differ in a specific exon, which is the alpha exon, and as you can see, this is really, really close to the MFGC binding site. So we think that these isoforms might actually have different binding partners and perhaps functions, and this is something we need to consider when we're actually validating the interactions. When I want to find out what the interactions are, I have to do some filtering. So as I spoke about before, we've got the IgG control, so basically the list of proteins here is compared to the list of proteins in the test, using a really cool bit of software called Saint Express, and this is basically a statistical method of probabilistically scoring the protein interactions. So basically, is it more likely to be a true interactor or some sort of contaminant or non-specific binder? This gave us a nice list of 110 proteins, but we hate lots of long lists, so we use a secondary filtering method called the crapome, which was kind of aptly named. So it's basically a site where they have all of the negative things that come up in negative controls of lots of affinity purification experiments. So if protein comes up a lot in there, it's obviously probably going to be some sort of contaminant. So we kind of calculate a percentage of experiments they turn up in. The kind of lower the percentage, the better. So this allowed us to cut off proteins that had more than 10% presence within that list. This gave us a nice list of 13 proteins, which allows us to identify some proteins of interest from there. We did look at both lists in terms of gene ontology and things like that, but this gave us some things to look at we might be interested in. This is just an idea of what the output from Saint Express looks like. Basically, there's just 2 main cutoff lines, so any proteins that sit above those 2 lines are ones that we think are important. So what we can also do is map all these interactions, the 110 of them, into string, which kind of looks like a little bit of a mess, but it's basically all the interactions that are previously known about between these proteins, so we can kind of see where things are fitting. We also compared that list of proteins that we got to known interactors of MFGC. A lot of these are done in muscle cells, so we didn't get that many known ones, but HDAC4 and 5 and MEF2A came out, and this basically just increases the confidence of our novel interactions. So the main kind of conclusion from all of this, based on some of the proteins that we identified, was that MEF2C has actually been held in a repressed state in these basal BB2 cells. So cabin is known to suppress activity of MEF2D, which is a different family member of the MEF2 family, which is identified there, and then HDAC4 and 5 are actually also known to directly repress MEF2 activity. So we think it's been held in this kind of similar state in our cells. But from both of these previous works, they found that calcium stimulating can actually release the repression of both of these, both cabin and the HDACs. So what we think is if we stimulate these BB2 cells with calcium, that we'll get some sort of similar relief of repression, and this may allow MEF2 to interact with other more interesting proteins. So we did this, and really nicely you can see that it actually reduced the abundance of both the HDACs and cabin-1, which just means we think they're dissociating from FGC after calcium stimulation. Just an example of what the same looked like from that analysis as well. Can't really see very clearly, but this is HDAC and cabin down here, so they're no longer significant interactors. We did compare both the data sets. There is some overlap, but also some new proteins that came out in the stimulated data set, but only really reflects a really subtle change to the interactome. We got some proteins that were interesting, but like no real kind of activating transcription factors or kind of an activating complex that comes in afterwards. So we are releasing the repression, but there's not anything really coming in to do anything exciting. So this allowed me just to revise the model of what we think is going on within BB2 cells with FTC. So obviously we've got this repression before the calcium stimulation, introduce calcium, and then it's taking both cabin and HDAC out of the situation. You still get HERA, which we got in both of them, that sits there, so it might be enhancing some transcription activity, but there's nothing really no coactivators coming in to do anything. What we also might think is that the repression is actually isoform specific, because I mentioned at the start they either have this alpha-1 or alpha-2 exon. And having the alpha-2 exon means that they're basically more resistant to this HDAC-induced repression. So it might be that carbon-1 is repressing the A2 isoform, whereas the HDACs are repressing the A1 isoform. But we need to clarify that in our validation. So just to conclude, so I presented the first co-IP mass spec interactome, of FTC in microglia-like cells. It's likely to exist as two main isoforms. It's predominantly been held in this repressed state by those three main repressors. Calcium can release MEF2 from this state, and it does allow it to form some new interactions, but as I said, there's no real new activating complex. So what we think is that calcium might be changing how MEF2C is functioning in different ways, and maybe it's meaning that it regulates gene expression differently, is it binding different places, so we're investigating this by the attack-seq. So future work, so the major pitfall of co-IP is that unfortunately a lot of the interactors you pull down might not actually be direct. They may be just cross-reacting with the antibody, be some sort of contaminant that's survived the filtering process, or they might interact with MEF2C but via another protein. So we need to validate all of those before we can really go any further. As I do all the fun genomics stuff, and then hopefully if science likes me, we can integrate all this data really nicely to make some really cool conclusions, just to help determine the role of MEF2C as a master regulator of AD risk, and then to bring it all back to AD, try and integrate some of this genomics data with AD-GUS data. Cool, thank you, happy to take any questions.
That was really interesting, and I think the calcium thing's really interesting, especially when you think about other risk factors like PLC2 we talked about earlier, and the calcium response there.
Yeah, so it, it actually sits at the bottom of that pathway that I think Emily and Eilish were talking about with PLGC2 and TREM2. I'm not an expert in that kind of area, but it kind of sits at the bottom where it integrates that calcium signalling, which means it changes the gene expression, which is why we think you might be doing something cool at that level as well.
Yeah, that's really interesting. Any questions?
You kind of stole my question. Did you choose calcium specifically to stimulate these in this experiment, or are you interested in other forms of stimulation?
It was mainly because all the previous literature said that calcium could release the HDAC and the carbon repression, so we thought that one would be a good one to try. Also, I was originally doing a different protein and nothing worked there, so I kind of had to go with all the FTC calcium stuff. So it's just mainly a happy coincidence, really.
I guess also on a related note, in neurodegeneration, is it known, I assume FTC leaves its inactive, its repressed state and becomes unrepressed. Is it known what that trigger is in the context of neurodegeneration, at what point it does it, how that works in disease?
Yeah, I don't think we really know much about how FTC is functioning. We don't know if it's overexpressed or whether it's whatever. We don't really know what's going on with it in Alzheimer's. So I think it's just part of what we're trying to figure out is what it's actually doing anyway, and what it might be doing in Alzheimer's as a result of that.
Sure, thanks.
I think everyone wants coffee now.
Thank you very much.
Thank you.
Okay, yes, that was the last of the ECR talks for today. I think we've got a coffee and poster break now for half an hour, and then it's on to the methodology sections. So I'm going to ask to say, if you're signed up for trial isolation, you're in the sun room, which I think is down there somewhere, and if you're in the omics session, you're in here. Excellent.
we hold off for a couple of minutes?
I should be ready in seconds.
Okay.
Okay. Do you want me to pause or go slowly? Sign. I don't know what the sign for metabolomics is.
Maybe we'll pause for like a minute?
Maybe we could Google the sign language for metabolomics.
Make it up.
Hopefully we can, there's enough on the slides that they can...
Probably, yes, yeah. So, oh,
Oh.
I've heard something, so. Can you hear us? Everybody's, yeah.
Okay, we're good.
We're good, okay, got the thumbs up. So broadly speaking, the field's developed into two different strands. So you've got signalling, so where people have been used to measuring low concentration trace molecules that perform some sort of signalling function. So I've picked out some neurotransmitters there, glutamate and GABA, cosinoids, IP3, diacylglycerols. They're in low concentrations, and typically this has been done in a very targeted manner, but also often in a quantitative manner to understand those signalling roles. Flip side of it is that there's also a group of individuals that have been interested in terms of metabolism and really trying to survey as many metabolites as possible as part of this approach. So like I say, we've got approaches where we can survey metabolites and lipids, all the metabolites and lipids that are present. So the spoiler in terms of this is that the answer is no, that we can't get a better, we can't actually survey all of the metabolites present at the moment in terms of chemical space. The reason why we can't actually measure all of the metabolome or the lipidome at the moment is down in this cube over here. So if we imagine all of metabolic space is represented by this cube, we might want to operate across a pretty large mass range, from 1 atomic mass unit up to 1500 atomic mass units, and across a pretty large polarity range over here. Now, most of our mass spectrometers can measure this mass range over here without too many problems. So there's been significant technology advances in terms of our mass spectrometry. We can do the polarity range as well, so we can measure it across different forms of chromatography. But the real killer is that metabolic space actually probably exists across a concentration range of 10 to the 9. So we're looking at things that are millimolar concentrations of bucket loads inside the cell, and things that are down there in femtomoles, so very low levels. And there's no one analytical tool that will cover everything. So if you're interested in things down here, you're much better off still using custom assays. But we're doing a better job all the time in terms of pushing down those limits of detection. Just to say something about lipids is that metabolites themselves are a very diverse range of molecules that we want to measure. So we go from things that are very lipophilic to things that are highly aqueously soluble, which is why we need lots of different chromatographies to separate those out. Lipids, on the other hand, though, they're a subset of the metabolome, but they consist of a series of basic building blocks, fatty acids. So just like you can for proteomics, and we're going to hear about proteomics in a moment, you can build up databases because you know that all your proteins have to consist of amino acids. We can do the same thing in terms of lipidomics. We know that all our lipids consist of fatty acids, and we can put together in silico databases in terms of those identifications. And that's been a huge help in terms of moving the field forward compared to sort of regular metabolomics.
I just realised, maybe just unmute yourself there just so that Rich can
where?
hear you. I just thought of that. He probably can't hear what you're saying. Sorry. Maybe Zoom?
Can they hear me at the back? And I've got a thumbs up back there.
I'll just for Rich. There we go.
Oh, okay. Oh, that better. Okay. So nobody's tell Rich about how rude I've been about proteomics and how it doesn't work. Okay. So in terms of doing, I haven't been rude at all. In terms of lipidomics and metabolomics, we've got different types of experiments that we can do in terms of it. So I thought I'd start off with what's often referred to as open profiling. And this has sort of been classically where we spent most of our time sort of developing methods. We're going to try and measure as many metabolites, or in this case many lipids, as possible and get a snapshot in terms of what's happening in terms of metabolism. Now this shows you some typical results we might get from a lipidomic experiment here. It's from a tissue extract, and along this axis down here, this x-axis, is our M/Z, mass to charge ratio. So this relates to the relative molecular mass of the species that we're detecting. And then over here is our chromatography time. And you don't need to be a mass spectrometrist to see that we get some nice separation between these groups over here. These groups are phospholipids down here, and these are triglycerides over here. We can collapse it down into a single chromatogram down here in terms of 2 dimensions, and you can see sort of how we can separate out, in this case, lysophospholipids, phospholipids, and triglycerides overall. Now, on mass spectrometers, if you spent a lot of money on them, we can actually work out a relative molecular mass to 3 decimal places, which freaked me out the first time I discovered that. So we can work out chemical formulas by the mass deficits that are present there, so we can begin to annotate in terms of chemical structure. However, if we really want to identify things, we have to smash them apart and do fragmentation in terms of our analysis. And this just shows you where we fragmented 2 different lipid species here. They're both phosphatidylcholines, but they weigh the same. So at least in terms of regular mass spectrometry, we refer to them as being isobaric, and we couldn't separate them simply by normal M/Z. So we smack them into an inert gas, and they fragment, and we can see the difference in the fragmentation patterns and identify those across there. So that's open profiling. And I thought I'd just sort of show you about how people have used this in terms of dementia research, in terms of open profiling. So we've all gone a bit sort of crazy and tried to do GWAS with metabolomics and lipidomics, so trying to do metabolomics and lipidomics at scale. And there are studies out there where people have done 25,000 samples using lipidomics. So I think one of the messages here is that we can actually do very large-scale studies if we want to, apart from you have a nervous breakdown when you're collecting all of that amount of data. This is a nice paper which came out at the start of 2020, looking at, it's effectively what was referred to as an MWAS, a metabolome-wide association study, looking at SNPs that are known to be associated with Alzheimer's disease and how they're associated with metabolites. So it's a Miami plot showing genome-wide associations of sphingolipids on the top, and then acyl carnitines and amino acids on the bottom down here. So you can do that across the genome. The big issue here is that we need large N numbers in terms of taking this type of data forward. This is a study that I've been involved in recently. It's not an Alzheimer's disease study. It's actually looking at myocardial infarction, but the data set is out there. And the top is from a cohort called PROMISE, which is looking at a Pakistan population, and down here is Interval, which is a UK population over here, and looking at associations of SNPs known to be associated, in this case, with cardiovascular disease, and seeing how they're associated with different lipid species overall. This is another MWAS-based study down here, looking at commonalities between the brain and blood serum. This is a study that came out of partly out of Rima Dawak's lab over in North Carolina, looking at commonalities between the 2 different data sets here. So they had a rather large brain bank that they could go and profile, and then look at some of the similar metabolite changes and lipid changes between the 2. And the interesting thing there is that we've seen a lot of changes in terms of bile acids, and bile acids have an important signalling role, but much better defined, I think, fair to say, in the liver than in the brain, sort of going forward. And they link this into cognitive outcomes, in this case cognitive decline. So if you're interested, that came out in scientific reports back in 2020. So I was told not to talk about my own research, but I've just slipped a slide in. So this is some large-scale studies that we're doing over at Imperial with the DRI over there, looking at 2 different cohorts called Airwave, actually 3 different cohorts, Airwave, Rotterdam and Finger, I flag it up because the data should be available for everybody. But what we're looking there at associations with SNPs that are known to be associated with Alzheimer's disease, and how different lipid species are associated with them. So this was looking at what ABCA7 might be associated with in terms of lipid metabolism. We've looked at several thousand people across the Airwave and Rotterdam cohorts, and then looked at associations between the SNPs associated with ABCA7, which lipid species are associated, and then also which other SNPs associated with Alzheimer's disease might be associated with those metabolites. So we begin to build up a profile going from SNP to lipid to SNP again in terms of mechanism overall. I'm now going to change tack though and just sort of show you that you can do it on cells. So this is an OPL-SDA plot. It's a supervised form of principal component analysis, looking at some cells that we got from Bart de Stupper's group, looking at ApoE and how ApoE affect microglia lipid metabolism. So each one of these dots represents a mass spectrum over here, and you can see sort of nice clustering according to genotype across there. We're looking at sort of 10 to the 5 cells, but I'll show you some cool stuff at the end where you don't need quite so much material. I'll go through this very quickly. This is just sort of hot off the press. We've been looking at activated microglia, activating them by LPS to see which lipid changes happen. Again, we've got an OPL-SDA model over here, so each dot represents a mass spectrum, and then we can go and interrogate our OPL-SDA plot to see which lipid species are most changed. And we've been particularly interested in a set of ceramides that have sugars attached to them, hexoceramides, and they seem to be profoundly changed during activation. But really the big message, I think, for this audience is don't be afraid of mass spec in terms of doing the analysis. It's just straight from the cell culture. So that tells you about open analysis, where I just try and detect as many metabolites as possible. It's a bit of a pain because you need good databases to do that, and we end up with a lot of unknowns as part of it. Sometimes you as PhD students and postdocs, you already know the answer to your experiment. So sometimes it's actually better to sort of test those questions. And here we have closed analyses or targeted analyses. So it's possible to target a particular pathway and get an answer out directly out from that. I think we're going to hear more about mass Westerns in a moment in terms of detecting particular peptides, but we can do the same thing in terms of small molecules. Look for specific small molecules and measure them in a quantitative manner as part of this approach. The classic way of doing this, if you're going to do it by mass spectrometry, is to use a triple quadrupole. Like Ron Seal, it does exactly what it says on the tin. It's got 3 quadrupoles in there, and the quadrupoles are used for selection. Quad 1 and Quad 3 are used for selection, and Quad 2 is used as a collision cell. What happens during this experiment is I send my ion into the mass spec, I select for it with Quad 1, it smacks into inert gas in Quad 2 and fragments, and then I just select for one of my fragment patterns. So I effectively have to have 2 pieces of information to get an identification. I can also do chromatography on the front end, and then I have 3 pieces of information. It's a bit like climbing. I've got 3 points of contact at that stage, so I've been pretty certain about what I've identified overall. This shows you where we've used something very similar to this for detecting eicosanoids. So it's a workflow that we set up for detecting eicosanoids, or actually oxylipins in general. So these are oxidized lipid species, where we could use it to detect different oxylipins, but also quantify them to understand their role in signaling pathways, particularly around inflammation. So I sort of flagged this up because I thought that might be interesting given the audience that's present here in terms of doing it. So I wanted to finish off though on another approach that you can use in terms of mass spectrometry. This is a bit more of a pain to sort of get set up, but it's very cool sort of, I think, once you do have it set up. At the moment, everything I've told you about has been involving tissue extracts or biofluid extracts or cell extracts. You can do mass spectrometry in situ. So this is mass spectrometry imaging here, and I flag up Zoe Hall, who was a postdoc in my lab, and now she's a new lecturer at Imperial College. So Zoe has been using multi mass spectrometry. There are other flavours for doing imaging, but I'll just go into multi. And you can take a tissue slice, You place it down on a glass slide. Don't put it near any fixative because that really dirties our mass spectrometer. We spray over a matrix to allow things to ionise better. And then this is the cool bit. We raster a laser across the tissue. And each dot of that laser produces a mass spectrum. So we get a whole series of mass spectra going across, in this case, a tissue slice from the brain. And we can begin to build up those patterns. And this is actually, I don't have one from the brain at quite this level of detail, but this shows you sort of where we're at in terms of technology. So this is from a liver tissue slice here, and you might not be able to see it with the lights outside, but or the light coming through, but we've actually got a 10 micron resolution here, so we're capable of doing single cell mass spectrometry as we raster across those tissue slices. In terms of single cell based approaches, we can also do it on single cells, direct from cell culture. Again, there's a fair bit of setting up of doing this, but this was a nice paper which came out partly out of the Cambridge DRI and partly out of Albert Kuhlmann's lab. We worked together at the Medical Research Council a little while back. So, but if you're interested, he uses an approach called laser mass spectrometry, and we'll take single cells and then get you a single cell, a mass spectrum back from that single cell as we go through it. So I think that's everything, and I'm going to hand over now to Rich.
So I think what we're going to do is hand over to Rich immediately and then do questions and discussion at the end, because we're hoping to kind of get a conversation going. So, Rich, are you ready to go?
Yeah, can you stop the screen share on this so I can do mine?
I can try, but I am not great at tech. And show.
And then
okay.
Then stop.
Oh. I'm so glad it's not just me.
Hopefully you can.
That should do it. Hang on. Can you see me?
Yes, it's... Yeah, okay,
Yeah.
we're good. Although you're not... Now you're full screen. Okay, have fun.
Right, perfect. Okay, thanks very much. So some of the things that Jules has spoken about with metabolomics are similar to what we would do with proteomics, but with proteomics we've got different options as well. So what I really wanted to do this afternoon was just give you a flavour of the sorts of methods that we can use for proteomics, because one of the things that we often get asked is which method's best for my experiments, and the answer to that always depends on what your experiment is. So at a very top level, you can use proteomics to answer lots of different questions. What we're going to focus on today is relative quantification of proteomics, so essentially spotting the difference in protein expression between two sample levels. Other people, you've just heard about imaging mass spectrometry from metabolomics. You can do that for proteins. There are ways you can use proteomics to look at protein structures, and that's quite interesting. There's some nice studies on sort of A-beta structure and protein folding using mass spectrometry. But for the purpose of today, we're going to go on measuring protein levels. And essentially, all the methods I'm going to speak about will fall into one or two groups. Either global proteomics, where the idea is to try and identify as many proteins as possible in the sample. And that can be whole cells, tissues, fluids, it can be subcellular fractionation. We saw a nice example of this before the break, looking at protein complexes, where the idea was how can I identify all the proteins in the sample. Or you can do it on groups of post-translationally modified proteins. So if you're interested in phosphoproteins or glycoprotein expression. And these approaches are great because essentially you're hypothesis generating, you're trying to measure as many things as possible, but these approaches do that in a concentration dependent manner. So as with metabolomics, any other type of mass spec, you'll identify the most abundant proteins in the sample, but you won't necessarily identify everything. The second type of experiment that we do is something called targeted proteomics. So here you know what you want to measure. So you've got a specific peptide or a protein or molecule that you're after. Again, you can do it on any type of protein sample as long as it's prepared properly. But the advantage here is because you're only studying certain molecules, first of all, you hit them every time. And I'll go on to explain why that's important shortly. and because you've got a reduction in noise, because you filter all the things you don't want, and you only look at these specific proteins, you get higher sensitivity, because actually it's not about signal, it's about signal-to-noise ratios within the instrument. So by reducing noise, you can see smaller signals. A lot of people think this is what mass spectrometry is like. If you've got a nice core facility in your university, you take a sample, you trot it down to the mass spec lab, they do some magic, and then 2 weeks later you get some data. Hopefully after this afternoon you'll have a bit more appreciation for the magic stuff in the middle. So whereas for metabolomics you measure a sample, you measure the metabolites, proteins are big, and proteins are not ideal for mass spectrometry. So the first thing that most often we'll do is digest those proteins into individual peptides. So from that we'll do it using an enzyme, usually trypsin, because trypsin cuts at known amino acids. You can predict what the peptides are going to be. For other applications you might choose to use different enzymes. And because we take what is a very complex protein mixture, so in a cell lysate there may be 20,000 different proteins, and then we fragment them into peptides, and if you get on average 40 or 50 peptides from each protein, all of a sudden you've got 500,000 different peptides. We have to separate those out before we analyse that mixture by mass spec, and we usually do that by HPLC. The HPLC is what we call lines in the mass spec, so the sample flows through the column, the peptides are looped off the end, and they loop straight into the little hole in front of the mass spec called the source, where they're ionised. So there's an addition of protons to each peptide, and that ionised peptide is then allowed to pass through the instrument. There are lots of different types of mass spec on the market. They all have slightly different layouts, they all have slightly different properties, but essentially they all work in the same way. So this is a schematic of a Q-TOF. So the first two, the blue and the purple bars here are quadrupoles, they act as mass filters. And then the big up and down thing at the end on this instrument is called a timer flight detector. This is what measures the mass. You'll see other instruments that are Orbitraps, they've got an Orbitrap on them which measures the mass. So different ways of doing the same thing. In any given experiment, we'll have peptides released out of the LC system, they'll go through the instrument, and we'll measure the masses of the peptides in the sample. And that will generate a nice mass spectrum with mass over charge ratio on the bottom and signal up the side. The fact that it's mass over charge is important, especially for proteins, because peptides can be +2 or +3 or +4 charges. And the same mass of peptide at a +2 and a +4 charge state will fly twice as fast. And so it will appear at a lower M over Z. So you need to know Z to calculate the mass of the peptide. Although that gives us accurate masses on the peptides, it does not give us identification. As Jules inferred from the metabolomics, these samples are complicated, and it's very easy to get different peptides with essentially the same mass. And so for proteomics, in order to try and identify the sequence of the peptide, we have a second step. So again, we've got the same peptides eluting out. This time in this blue quadrupole, it's acting as a mass filter. It gets rid of all the peptides we're not interested in. It selects one peptide, the green one. That green peptide collides with a nitrogen gas in the collision cell to generate fragments. You measure the mass of the fragments, and you generate a mass spectrum from that individual peptide. And the sequence of fragments you get, essentially, you can read off the amino acid sequence because of the fragmentation pattern. You'll hear terms in mass spec labs in the literature about MS and MS. So measuring the mass of stuff is MS. Doing this fragmentation of identification is MS. You've got two mass measurements, one in that first blue quadrupole where you're isolating the peptide of interest, that's one mass measurement, and then the second mass measurement is of the fragments. And this is a fantastic tool for giving protein identification. This happens very, very quickly on modern mass spectrometers. You can fragment up to 20 peptides a second and get really good data. So in an hour-long gradient or an LC gradient, you can identify hundreds of thousands of peptides. But in and of itself doesn't give you any quantification. And there are lots of different ways to get quantification of peptides between samples. And I'm going to go through the three major ones now. The first is what we call MS1 DDA. There's lots of acronyms in mass spectrometry, so pen and paper time, I'm afraid, for those. DDA stands for data dependent acquisition. And essentially, this is the same as the profiling approach that Jill showed before, where we take samples that are prepared sequentially, trypsin digest, LCMS, you generate a profile. This is the MS1 profile. So this is the mass of all the different peptides in sample A, masses of all the different peptides in sample B. You align those and you essentially do a spot the difference to work out which peaks are higher in one sample than the other. So in this case, we see a down-regulation of the yellow peptide, therefore the yellow protein in sample B, and an upregulation of the blue one. The advantages of these types of label-free approaches is that there's no limit on sample numbers. One of the downsides is you need good reproducibility of your LC separation and instrument sensitivity, because you're trying to compare from two different runs. If your LC separation is different between experiments, it's hard to align the peaks and say this peak in sample A is the same as this peak in sample B. You can do MS within this, and that's why it's data dependent, because the MS that you do is dependent on having the data for the mass of each peptide. That will give you identification of the peptides. And we'll compare that shortly to a new method called SWATH, where we don't do data dependent, we do data independent. But really, MS1 DDA has kind of been the mainstay of quantitative proteomics for a number of years now. You have got an option to use tagging methods for quantifying proteins. So I'm going to give an example here about isobaric tags. There are two flavors of isobaric tags, one called iTrack, which is on this slide, and one called TMT, which is the same thing but generated by a different vendor. And in these experiments, you take your samples. For iTrack, you can take up to 8 different samples. For TMTs, now you can take up to 16 different samples. You label each sample with a different tag. The iTrack tags are shown at the bottom. Essentially, the overall mass of all the tags is identical. That's why they're called isobaric tags. But the mass distribution across the tag is different. So you can see here we've got different masses in that reporter group and that balance group. The protein reactive group binds to at the N-terminus of every single peptide. So you label every peptide in the sample, you mix them together, analyze that mixture by LC-MS. And now, because each peptide has the same mass tag to it, you can't distinguish in the MS1 where that peptide came from. So essentially you see a stacked bar here where the blue, red, yellow is the same peptide with a different tag on it, but they have the same mass. However, when you do the MS2 and you fragment that peptide, you also fragment the tag. That releases this reporter group. The reporter group and the different tags are different masses, and so you can see those released to the top. That gives you relative quantification of that peptide across the 8 or 16 samples, and the peptide fragments to allow you to read off the peptide sequence. And so again, this is a nice way of generating relative quantification data for lots of peptides. So hundreds of thousands of peptides from maybe 4,000 or 5,000 proteins in the sample. There's an advantage here because samples are analysed simultaneously. Because you mix the peptides, you can do things like further fractionations. You can isolate certain types of peptides from that mixture. But you are limited to a smaller number of samples per run because of the number of tags. Another advantage here is you don't get as many missing values. So I'll speak about missing values shortly again, but because you're fragmenting the same peptide at the same time from all 8 samples, you get data from all 8 samples. And generally these are more expensive approaches because you've got to pay for the label and you do take a bit more instrument time. The final global proteomics type method that we use is something called DIA, data independent analysis. So an example of this is SWATH-MS, which we use in my lab, but other vendors have their own tools, all called different things, but essentially the same thing. Here, instead of selecting an individual peptide for fragmentation, you select all the peptides in a range called SWATH. So you might choose a 5 Dalton window and you'll fragment all the peptides that were eluting in that window at that time. Then you'll go 5 Daltons up, 5 Daltons up, 5 Daltons up, and you'll scan the mass range in 5 Dalton steps, fragmenting those peptides each time. And then you'll go back to the beginning, you'll do it again, you'll do it again, do it again. The advantage here is that rather than data-dependent acquisition, where you only fragment the things that the mass spec chooses, here you fragment everything. So every visible peptide in the sample, you will fragment every time because of the way that this is selecting ions for fragmentation. And this helps generate what we call digitized proteomic maps. So it's a map of essentially all the peptides within the sample. And it's much less biased. You get much less missing data out of this than you would a standard DDA approach. Because with DDA, if you don't fragment a peptide in a sample, you don't get any data from it. Whereas here, you can go back and you can reinterrogate these maps and you can fill in the gaps. And by comparison to sort of pre-prepared reference libraries of all the peptides that you know should be in the sample, you can interpret these relatively straightforwardly. This is a new approach that's really been around maybe for the last 3 or 4 years now, used in anger, but it's certainly taken over, I think, as one of the main ways to do global proteomics. So which one do you choose? Obviously, every method's got its own pros and cons. The MS1 DDA is established. It's relatively straightforward. There's no limit on the sample number, but you do get missing values, probably more from anywhere else. And you can't extract more data from it. If your peptide hasn't been fragmented in that sample, you don't get the data. It's essentially gone. For isobaric tagging, it allows you more flexibility in the sample prep. You can do peptide enrichments, and you don't get as many missing values. You generally get a lower false discovery rate in terms of changes. So the stats are slightly better on this because you're doing everything at the same time. Although here you've got a limited sample number, and the data processing can be quite chunky. But so DIA approaches are probably coming into their own now as we develop the software and better ways to analyze the data. In our lab, we generally get better coverage of the proteome with DIA compared to DDA, although not as much as with isobaric tagging. But the ability to go back and re-extract more information from the data is a real positive, I think, for this type of approach. So they're the 3 global methods that you'd want to use. Jules mentioned previously about targeted quant, selective reaction modeling with triple quad. So I'm not going to go through this again. Because essentially it's the same as Jules described. We've got peptides coming out of the LC column. You know what you're going to measure. So you select the peptide that you're interested in, fragment it, and then select one of its fragments. For proteomics, we can measure multiple fragments from a different peptide. So we get specificity by measuring 3 or 4 or 5 different fragments. Because they're from the same peptide, you know that they correlate. As I said before, the signal, you don't get a nice mass spectrum here. You get this is the number of counts hitting the detector from things of mass 763, which have a fragment of mass 624 at that given time. But over an LC gradient, you can generate nice clean peaks with virtually no background, and that gives you more sensitivity. From a quantitative perspective, these are also more linear. So SRNs are linear across about 5 orders of magnitude. And if you have a heavy isotope labeled version of the peptide that you're interested in, you can spotlight the sample at a known concentration, and you can look at the ratio of the heavy peptide signal versus the endogenous light unlabeled peptide. And you can use that to generate absolute quantification. So what's the concentration of this particular protein in my sample in micrograms per mil, or nanograms per mil, or nanomole if you're working in plasma, or copies per cell if you've got cells and you know how many cells you've got to go into the experiment. So this is really quite powerful. Essentially, it's a mass spec Western blot or a mass spec ELISA. But it has several advantages over ELISAs. First of all, there's no reliance on antibodies. So you know what you're analysing because you can characterise it well. With an ELISA or even a Western blot, you get additional bands and you can never be 100% certain how specific your antibody is. You can also multiplex. So these MRM transitions you can do in about 50 milliseconds. So you can measure lots of peptides and lots of proteins in a sample in a single run. So if you've got an experiment where you want to measure 10 or 20 proteins, rather than going by 10 or 20 ELISAs, you can set up one mass spec method to analyse all of them. They're probably equal in sensitivity now to antibody-based methods. You definitely get a greater dynamic range in terms of quantification. As I said, you can use a heavy isotope standard, which costs £200 or £300, and spike that into your sample to get concentrations. However, if an ELISA is available for your peptide of interest, I would still use it because establishing these methods takes time. You need access to expensive kit, you need specialist expertise to run them. And so in many cases, especially for sort of plasma analysis, then ELISAs are probably the way to go. But if you want to measure things like splice variants, post-translational modifications, or things where you've got no antibody, or the antibody is rubbish, more to the point, then then MRM is very definitely a good option for doing this kind of targeted work. So I'm going to leave that there. It's quite a quick whistle-stop tour of the main methods that we use for proteomics, but hopefully it's answered some questions, and hopefully it'll help to kick off some kind of discussion.
Cool, thank you both. Those were really great talks. Yeah, I was just trying to work out how we're going to do that.
Do you want to sit down as well?
I think we'll be too low.
Oh.
We can hover, right?
Yes, yeah.
We're great at hovering. So I have written down some questions that people submitted in advance, but I'd rather we have a kind of natural discussion because I'm just the ECR at the front. So if you guys have questions, first of all, it might be a good idea to have a show of hands if anyone's likely to be doing any proteomics, first of all. Okay, no hands went up, Rich.
We've got one.
Oh, one hand, okay. Okay, and yeah, some people have done it. Okay, so we've got like a couple. And then anyone, yeah, anyone doing any metabolomics, lipidomics?
Planning to.
Okay, cool, okay. So does anyone have any questions that they feel brave enough to ask right away? Emily, go for it, and be loud, or come up.
So we was going to do some lipidomics and some samples, right? And the initial idea, because I don't know that much about lipidomics, was to do like a global kind of screen, like non-hypothesis driven screening. But then we were advised by someone that doing a global screen like that is going to result in lots of problems, and therefore that we should do like targeted kind of bits on groups of lipids instead. But then there's also kind of like still a pull within me to do the whole global screen. So I just wanted to know what your opinion was about the different approaches.
So I think it depends on what you want to measure and why you want to measure it. So, and it goes back, for the lipidomics, it goes back whether you're interested in signaling or whether you might be interested in metabolism changes. So for signaling, I think it probably is better to be targeted just because some of the things that you're interested in are so low in concentration. PIPs came up earlier and they stick to everything as well, so they're a nightmare for doing chromatography on, and they're there in low concentration. So for those, it would probably be better having a targeted analysis, and that's why we did a targeted analysis for the eicosanoids, because they have the same issues. Whereas if you're interested in metabolic changes, so we were hearing about how cholesterol metabolism was involved in multiple sclerosis right at the start, then actually the global profiling can be really useful because you not only detect cholesterol, which is there in high concentration, you also detect some of the lipids that are associated with cholesterol, some of the precursors, cholesterol esters, and then potentially some of the other things that might go through those transporters as well. So I think it's having that sort of, discussion at the start about what you want to get out of the results, and then perhaps designing the assay, and then potentially doing both, so you can get very complementary results from doing the two sets of assays, and it's not unusual for us to do a global profiling to work out which bulk lipids had changed, and then go in with a more targeted analysis of the eicosanoids. Can't do the eicosanoids for things like imaging because they're there in too low concentration, but you could design it for the LCMS-based approaches. So I'm not sure whether I just sat on the fence there.
Well, I think it's good. We're comparing like to like a new Alzheimer's cell line, the high and low risk lines.
Yeah.
So I think the global one is maybe the way.
So I would sort of start with the global one, and then once you know what's changed, you've got two possibilities in terms of lipidomics. If you've done a good job of measuring that lipid class, you can strip that data out of your original data set and re-data mine it. Or you could then set up a targeted analysis and do a more quantitative approach. The advantage of going that way around is you know what you want to measure, and then on some of them aspects they actually come with some of these sort of targeted analyses sort of set up already. So I know one of the companies has done quite a lot of work, Waters, have done quite a lot of work on in terms of some of the targeted analysis for lipid species. So yeah, I probably, from what you've described, go for the open profiling in there.
Thank you.
Have we got any more questions from you guys so far? So a couple of people have asked whether it's possible to do lipidomics and proteomics on iPSCs, which I'm putting to both of you. If you could both just kind of talk about the numbers, but then one of the questions was about how to normalize across experiments. So for example, would you use cell density, or an internal lipid was suggested, or a different metabolite as a control, like an equivalent of a loading control in a Western blot, I think is what they mean. Yeah. Rich, do you want to go first, and then we'll go to Julian?
Yeah, because that's a really complicated question. So we've thought about this quite a lot. So generally, if you can be fairly confident that there's nothing significant between your samples in terms of total protein concentration, then we tend to normalise by total protein load. So you may end up with different numbers, because when you extract protein from cells, you'll do that at a different efficiency. You'll get some variable losses perhaps through that process, and so we end up, usually if you start with the same mass of tissue or number of cells with sort of 20 or 30% normalisation at the end of one sample to the other. And the way that most of our software does that is it assumes that most proteins don't change, and so we normalise all the data to essentially the median within the data set. That works fine until you're doing things like samples where you're doing phosphopeptide enrichments in a sample where you turn the kinase on, and actually you've got far more phosphopeptide in one sample, at which point we'd probably flip over to doing total cell number and hope for the best. But generally we would do it. There are a couple of people who've used something like individual proteins, so histones have been suggested as a decent internal control for protein, because you tend to always see them and they tend not to change very much. But histones are quite complicated and they get modified, and I've yet to be convinced by any individual protein which is a good loading control. Definitely not GAP-DH.
Okay, and is there
I was going to say what Rich said.
Great.
We have the same problem with lipidomics and metabolomics, and we normally try and normalise the total protein content in a lot of cases. Where that's difficult, you can also normalise to cell number if that's easier to do, and both those things work. And then we have very similar issues to what Rich has just said in terms of sometimes we can normalise to the total amount of metabolites that we detect, providing there isn't a big change in one of those metabolites as well. So to go back to the question about whether you could do proteomics and lipidomics or metabolomics on the same samples, the answer is definitely. And you could also do transcriptomics as well. So if you think about the workflow that you use, you're often throwing away one level of organisation, as it were. So we'll do a cell extract and get a protein pellet, and that protein pellet you could do proteomics on. You have to be a little bit careful that some of the older extraction procedures, things like perchloric acid, will actually nicely oxidise proteins, and in the days of Dye gels, we used to have lovely tram lines across some of our Dye gels, but they're complementary. And if you look at sort of how you isolate your mRNA as well, you're usually throwing away the small molecules, so it's possible to do that and get direct information. Probably easier to do the metabolomics, lipidomics and proteomics all together.
Okay.
I think was there an iPSC question as well, Megan?
Yeah.
Yeah, so we've done proteomics for iPSC before, it's fine. The protein amount you need to do is relatively low, so 10 to 50 micrograms of proteins, you're talking less than a million cells.
Okay.
Generally, so if we always tell people the more protein you've got, the better, because it's easier for us and you're going to get more peptides, more data. But actually, if you can generate a couple of micrograms of protein, then you'll probably do okay. The difficulty with iPSCs that we found is actually getting them grown properly, because they keep changing in culture. Not like HeLa cells that just stay, relatively constant. iPSCs are constantly developing, so actually the challenge is more on the cell culture side, and making sure you've got harvests at reproducible time points. If you start to differentiate them rather than harvest straightforward after that.
Okay. Nina has a question which I suspect will be related.
Do you want to maybe come up just to make
Yeah,
sure Rich
or I
can
can
hear?
repeat it.
Yeah.
So I was just thinking, when you run the essay, you say most of them are quantitative, right? So do you need to know in the beginning what your reference sample is, and the process will make it already quantitative, or do you get kind of like a raw count, and then within the analysis you can still define kind of what type you want to use as a reference to normalize? Because I think I wanted
This is
to...
a lot to repeat. You're going to have to come up with it. If you're aiming it at Rich, then
I only thought about half of that as well.
Yeah. Sorry, Nina.
No, this is also super specific for what I want to do.
That's the point of this. So
I want to
that's
do proteomics, but I'm going to have so many different groups that I think in the analysis I want to maybe do different groups as my reference to see how are the quantitative changes. And you said most of these techniques are quantitative, so am I getting something like a raw count, like RNA-seq raw count, and then I can do it, or is it more like I need to define in the beginning what will be my reference?
No, for either, whether you do label or unlabeled, you essentially can generate multiple comparisons of an experiment using multiple references. And it does mean that you've got to run the same part analysis again, but it's not too difficult.
Okay, cool.
So we especially for label-free approaches, used to extract raw areas.
Okay. Do you have any follow-ups?
No, that's fine.
I can put you in touch if you want to.
Yeah, that's the thing. I think I'm not concerned about running the experiment. I'm more concerned about the data I'm getting back and then how I am supposed to analyze them.
We have a few questions on that.
Yeah, and if you're going to do something where you've got multiple comparisons like that, as long as you make sure you've got all the conditions in the same run and you randomise them appropriately and things like that, which designing those experiments is again a bit of a challenge to make sure that you're not biasing within your batch structure, but yeah, you should be able to, you can do multiple comparisons from the same block of data, that's not a problem.
That kind of leads on to a whole batch of questions that I've lumped together. You're dismissed. Thanks, Nina. Thank you. So we've had quite a few questions about how to approach the analyses. So one of the questions was how do you analyse quantitative proteomics data, which I suspect might have been you.
Can we have another day for that?
I think it might be worth just sharing contact details and stuff.
I've got a quick suggestion if anybody wants to get involved in processing data. There are these things called galaxy workflows, which you might have come across. So there is a galaxy workflow proteomics. This is assuming you don't want to put, some great vendor solutions, and some people have produced some really good open source stuff within R. But I don't know Galaxy P so well, but I know the equivalent in terms of metabolomics. So the big advantage of it is it's all free, and then it becomes quite push button over there. And if you are doing metabolomics, I've got some French colleagues who managed to convince the French government that it was a good idea to set a server up called Workflow for Metabolomics, and everything is effectively sort of push button in terms of the analysis. So I think if you're starting from scratch, that's not a bad place to go. And for the French metabolomics community, they have workshops in both English and French, so depending on how good your French is in terms of that. And I know there's a similar thing for proteomics, but Rich might know more about how well that works. It just seemed like quite a good solution if you don't have the software in your own lab. So we partly go from vendors' software and then into our own written stuff, and our own written stuff can be a bit clunky at times.
I already
I think that sounds language.
Sorry, Rich, what?
That's pretty much the same for the proteomics data. So most of the vendor software, all the mass spectrometry you come across will have a workflow, and that workflow will at the very least take you to the point of knowing what peptides, proteins are in your sample and some measure of intensity. And so the guys that work with us tend to, some people just take that data and analyse straightforward ANOVAs or whatever, and then go into doing pathway analysis to identify groups of proteins which are changing in concept. Others will spend a bit more time in R, but again, there are dedicated R packages that people have set up now for analysing these data workflows. So I think new students that come into us to do proteomics, I just tell them to go on a standard R course, learn how to use R, and then basically there are packages out there for people to get involved with. But it depends how deep you want to go into it. You can do an entire PhD if you wanted to, analysing a mass spec data set in depth. Or you could do a pathway analysis, identify 3 things of interest and move on and do some biology.
Or you can do what I've done and get some funding from the DRI, have a collaboration and go, I don't really know what I'm doing.
Get me.
These are some really nice things. Please can Rich and Anna do all the complicated stuff for me, and these are the questions I want to answer. I highly recommend that route. So some people have asked, we kind of already talked about this, are there computational tools to analyse and combine the different sorts of techniques, so the lipidomics, the metabolomics, the proteomics, and specifically they mentioned AI, and are there practical courses? So are there any softwares or anything that you'd like to mention aside from what you've just kind of spoken about, because it's kind of related? But Rich, got anything?
No, the best thing that we would say is if you speak to the vendors have usually got online courses. So we do a lot of work with SciEx, and the SciEx have got a thing called SciEx University, and you can do courses on how to run their mass specs, but they'll also give you courses on how to use their data analysis software.
Am I the only one here who doesn't know what they mean by the vendors?
Oh, the people that sell the mass spec.
People who sell mass spec.
Yeah.
Okay.
We're a bit funny as a community in that we make the distinction between industry tends to be pharmaceutical industry, and then we have these mysterious people called the vendors who buy us drinks at conferences. They're sort of halfway between the two. Yes, yeah, they can get you into lots of trouble, the vendors.
Okay.
And that's what they're most useful for, to be fair, sometimes.
So, but are the people who are here, are you going to be doing the mass spec yourselves, or kind of sending stuff off to other people to do? Okay, so it's probably more working with collaborators.
They told me they give me the data, and I don't know which version, and then I keep analyzing from that point.
Okay.
But I think they pre-process them, hopefully.
Okay, so hopefully it's pre-processed.
Yeah. I was going to say, as a general rule, and I'm sure, hopefully Rich will agree with me, find somebody friendly that you can collaborate with, so you can actually sort of pop around there and have those chats and things. And then don't assume that we actually understand everything in terms of your model. So sometimes it's really handy for us to understand why you're trying to do the experiment, because we might then redesign how we're doing the analysis. Even at the point of the mass spectrometer, going back to the question around targeted and untargeted analysis. If there was something very specific you wanted, it suddenly gets us thinking in a different way in terms of which mass spec that we might use, and definitely for the data analysis. In terms of combining data, there are various people that are using all sorts of artificial intelligence-based approaches, but I would probably recommend using something quite simple at the start. There's a thing called multi-block PCA, so it's a multi-dimensional version of principal component analysis. And you can nicely begin to combine data in there, and it's visual, so you can actually see what the patterns mean, whereas a lot of other tools can be quite black box, so you don't quite know what's happening and why you've got those associations. So I like to sort of see things and sort of how they happen, but yeah, that would be my recommendation in terms of an approach.
Have we got any questions from online?
No.
Okay,
no questions.
That's cool. I'll just do these ones because there are a couple that I think would be quite quick questions. One is it possible to do lipidomics or proteomics, metabolomics on isolated brain cells from quite small regions? So I imagine this person is thinking about isolating cells from like cortex, hippocampus, that sort of thing, and then doing the omics. Would you need to pool samples in order to just get enough stuff?
I think in the first instance, most of the time, Rich has already said this, we're set up for doing quite bulk analysis, and it's easier for us to start off with around about 1 million cells or so. That said, I showed you some examples of where people are doing single cell analysis. So the mass spectrometer itself is so sensitive, it has no problems sort of detecting metabolites at that level. And it's really how can we handle small amounts of material. And that's what I found is the big challenge. So it depends on how much time you wanted to invest in that. If this is going to be, sort of a 6-month, year project, then actually, you can go down to really quite small numbers in terms of material.
As a follow-up to that, can I ask about like human post-mortem tissue? Is there a problem if the tissue is dead before you get the sample?
Only in terms of, the usual stuff is that, how well does that represent the biological processes you want to measure?
Yeah.
So some of our small molecules go off very quickly.
Yeah.
The nice thing is that we actually know how quickly they go off. So some of them are used for working out time of death. So there's a lab out in Russia that's actually using brain tissue to work out by metabolomics, work out how long a body has been left out. I don't know what they're up to. So you have, and for lipids, certain lipids are more predisposed to oxidise than other lipids, particularly around the signalling side of things. But there's no reason why you couldn't do brain tissue, and we've got one project looking at brain bank to do just that.
Okay. Oh yeah, follow me, go for it. Oh, sorry.
Is it like the fixative that you use to fix the sample or anything like that?
It does for us. So a lot of the fixatives that people use, when they do, particularly around histology, they fly very nicely in the mass spectrometer. So we end up detecting the fixative and not the things that we're interested in. And then worse still, they can end up getting everything dirty. And I'm fine if I've got the front end of the mass spec that's dirty, but the further it goes back in the mass spec, the more and more trouble that we're in. So actually, for a lot of the metabolomics and lipidomics applications, we just like to either have fresh tissue, or for the imaging, they're just put straight out onto a glass slide. There are some different forms of mass spec where you can, so some of my colleagues that do DESI claim that DESI will not be affected so much by fixative, and we can subtract out some of those contaminants. But actually it's easier for us if they're just not there in the first place.
Yes, there are good methods for measuring proteins in FFP tissue, formalin fixed, paraffin embedded, essentially by going through the dewaxing process, and then the fixatives all work by cross-linking proteins, which is generally a bad thing, but there are chemical methods to uncross-link those proteins. Your recovery tends to be a little bit less, and the data tends to be a little bit noisier, but it is possible. There's quite a few people now have done proteomics on FFP tissues. That said, if you can get a fresh protein, we'll use that.
Palomi, is it a long one? Do you want to come up, or is it?
No, it's not. It's very short.
Okay.
So you mentioned something about mass spectrometric imaging. So what's the kind of material that you need, and how do you identify individual cell type, or how would you do that?
How would you do? So this is Rich. This is a question on mass spec imaging. So shall I'll go first with the metabolomics, and then you can say.
Yeah, go for it.
So it's actually just the same as you would with a histology slide, but we would just take, so on the microtome, we just take a frozen slide that goes straight onto glass, and then we would image by MALDI. We raster a laser across there, and then we get a whole series of mass spectra. Now, if you're taking a whole series of slices for imaging, you could then use one of those for histology. So you can work out where the cells are, and then you can work out where your metabolites are. And there are people that have developed software for aliasing the two images together, so that becomes really quite powerful then. And you can begin to see which cells that you're dealing with. And then certain cells you begin to sort of realise have their own metabolic profile sort of associated with them. So you can also use that. The big issue though is one of time, so it takes quite a while to set this up as an approach. And then each one of those points have to be collected. So you can be acquiring an image across 3-4 hours in some cases, but mass specs are getting quicker all the time. So.
And so this technique is purely for the frozen sections or histology. Nothing would be done with co-cultured cell, for example?
People have actually, so you don't have to freeze them. So you can get some mouldy plates now where you can put individual cells and do the mass spectrometry on there, and then people are building mass specs that go in conjunction with FACS sorting.
Maybe for live cells.
Well, how alive they are on the mouldy, they're definitely dying on the mouldy plate. But you could at least get a profile then of the cell that you're interested in looking at. That's okay.
Okie dokie. A probably, hopefully quick one, because we're over time, but I feel like there are still questions, so we should keep going. How many proteins would you expect from proteomics on cell culture supernatant? Is that an answerable question?
No, because it depends on what the cells are kicking out. So we've done it before, and we can get up to 1,000 proteins in cell culture supernatant without fetal calcium in it.
Okay.
So in terms of secreted proteins, but it really does seem to be sort of system dependent.
Okie dokie. And a methodological question. What is the best way to isolate cells to maintain the lipidomic / proteomic profile? And then someone else has also asked if you could comment on the effect of temperature, because some of these, certainly I do microglial isolations, you have to kind of heat the enzymes up to 37, but then you want to keep everything else cool. So can you comment on the effect of temperature on both lipidomic and proteomic profiles? Rich,
do it quickly.
Do it quickly, okay.
Do it fast.
And do it cold?
So we're always concerned about sort of changes once you've started to mess around with the tissue and disaggregate it and add stuff to it. So there's a group in Denmark, who's a phosphorylation specialist, and he did a set of experiments where he was taking cell culture samples and then essentially utilizing them in boiling SDS just to try and stop everything in one go. And he showed that essentially at 10 seconds, if you were 10 seconds too late, you could see differences in the phosphorylation status, which is a bit scary, really. I think you've got to be practical about these things, making sure that you're doing things on a routine timescale. So if one sample takes you 10 minutes, the next one takes you an hour, you can expect to see a loss of reproducibility if you're doing that.
Okay.
So we would generally say keep doing things as quickly as practically possible, as on ice as practically possible, but consistency is key.
Okay. Do you have anything to add to that?
It's exactly the same for us. Yeah, do it quickly and then get it cold.
Okay. And then that one. One question here that the reason I've left it till last is that I've not been sure how to phrase it, is about how to present omics data sets in an unbiased way that is not just in support of your hypothesis. I'm not sure how to word that, because it was...
How do you not fiddle your results?
Yeah, basically. How do I not go to Rich? I've got all of this stuff. So Richard and I have smushed up some brain regions of an amyloid model, and we're going to come up with a list of a ton of proteins, and I could very easily go, well, C1q is actually in that list, and that's great for me because I work in a complement lab. So now I'm going to go and talk about that for half an hour. So any comments on how to present your data in an unbiased way?
Well, I think the multivariate statistics are actually quite good because they take into consideration the whole data set, so you can then begin to summarise it. I guess the other thing though to do, in terms of sort of wider reproducibility of results, is that we're all being encouraged to make our data available. So when you do come to publish it, maybe there's a better way of processing that data, but if it's out there online, then somebody else can come back and say, look, I've reprocessed it, and look, I found all these other things involved in complement.
Yeah. And then there's also you might not want, as long as you're not lying about what your data is. Like I could say, well, this was upregulated and this is my area of expertise, and so that's why we're following this up. So it's not illegal to follow up your story of interest.
Yeah, I guess the other thing is get good at false discovery rates, because, you know, just like transcriptomics, we've got a high false discovery rate. So.
Okay. Has anyone else got anything else? So are both of you happy to make your slides available, if anyone, because there are quite a few references, maybe not the data if you want to get rid of the data, but there were some references that I was trying to jot down. I was like, no, I'm not getting there quick enough.
Yes,
and then, are you coming to the rooftop?
Oh, I didn't know there was I might have a quick beer if that.
Okay, so if anyone's got any questions that they don't want to ask in this sort of setup, then we're having drinks on the rooftop. I'm really sorry to those of you who are not here in person. But
I'm going to go make a cup of tea now.
Don't do it on your roof. Okay, so thank you so much for joining us, Rich and Jules.
Well, thanks very much.
And cool. Thanks, guys. Yay!
Right, we'll just stop the Zoom.
I don't know where anyone else is in order for us to